and then fine mapping of RF4 and RF3 for fertility restoration of WACMS in rice. Then identification of candidate gene for rice improvised virus resistance UPL. Then identification of 3 genomic regions associated with zinc uptake in rice grains. And functional validation of identified candidate garbage resistant genes. So with this brief, now I invite Dr. C.N. Neeraja to speak on the topic, the approaches in biofortification of food crops with the nutrients. Now over to the guest speaker. Madam, please carry on.
Madam, should I cut short my presentation or should I restrict it to 45 minutes?
Yeah, madam, you try to complete by another 30 minutes now.
Okay. I shall be quick because we have lost.
Next speaker is also waiting. So
yeah, sure, ma'am. Sure. I'm just sharing my presentation.
Okay.
Are you able to see my presentation?
Not yet. Not yet. Okay, now it's.
Are you able to see?
Go to slide view.
Yeah. Okay. Good afternoon, organizers of this workshop and conference. And I'm thankful to the organizers and welcome to the agricultural graduate students for attending this very useful conference in the backdrop of COVID. The topic given to me is approaches in biofortification of food crops with lot of nutrients. As you are all aware, food security is already we have attained because of green revolution since 1960s, and it is different from nutrition security. As you can see, the food production is ample and the access is provided through public distribution system. However, the nutritional availability is not expected as it should be because we are solely dependent on cereals. So we are lacking in balanced diet, leading to micronutrient malnutrition. So more than half of the world's population, especially from developing countries, suffer from this micronutrient malnutrition because we are excessively dependent on cereals, and the crucial micronutrients like iron, vitamin A, calcium, iodine, zinc are more critical for responsible for this micronutrient malnutrition, which is also known as hidden hunger especially. So this increases the disability of adjusted years, so indirectly impacting the quality of life and impacting the economy of the country and productivity of a person. So the malnutrition, as I said, it leads to a lot of economic loss also across the country. And as you can see from the figure, the global hunger index 2020 has clearly shown India ranks very dismally in the global ranking for hunger index. So 1/3 of the world's malnourished people live in India, and even the global hunger index severity is also almost serious to alarming. As you can see, the recent survey of NIN, when they have done around 1 lakh children, at least 35% of the children are stunted, around 22% are underweight, and anemia is there at least in 40% of preschoolers. And if you see, 40% of female adolescents have anemic conditions, means they are not having required hemoglobin, and their mineral deficiency, as you can see, they range up to 32%, vitamin A 22%, vitamin D 24%, and all lot of mineral deficiencies are there. And we know very well why this much mineral deficiency across the population is because of the poverty, poor purchase power, social and economic equality, as you can see, females or girl childs are more susceptible to these deficiencies, and as I was been telling, excessive dependence on rice and wheat. So what government is doing? Government is having lot of ways to combat this malnutrition, including the Food Security Act. There are public distribution systems, midday meal schemes, and ICDS, National Nutrition Mission, and recently Dr. Professor Nirmala Sitharaman, our Finance Minister, has announced the mission portion also, merging the supplementary nutrition and portion abhiyan. So, and the ways to combat micronutrient malnutrition are food supplementation. As you know, this government provides iron, folic acid, zinc, vitamin A, and under Niti Aayog and National Nutrition Strategy. In addition to this, recently food fortification is also there with the standards following by FSSI in wheat flour, rice, oil, salt, and with various minerals and micronutrients. And everybody, all of you know about iodized salt, which is a success story. So recently during August 15th, Honorable Prime Minister has announced the fortification of rice distributed under various government schemes, including PDS and midday meal schemes. So what precisely they will be doing is they will be adding fortified rice kernels with the 1 kg of regular rice. So, but what is the scaling? If you see, in PDS we need 328 lakh tons, but we have the capacity only for 60,000 tons. So the disparity is more for biofortification. It is good that we can address the malnutrition at least vulnerable districts, but biofortification, which is an agriculture research intervention, could be the ideal solution for addressing the malnutrition of the country. So what is biofortification? Since most of you are the students, you might be hearing about biofortification during your classes, or you must be coming across through various research papers. So biofortification is strategy of genetic enhancement of staple food crops with enhanced nutrients, and it's the most economic and sustainable way of combating malnutrition problem. Why? We will see one by one. There are 3 approaches of biofortification. One is first is the agronomic, where you will give external application with appropriate nutrients. Then genetic modification technology. As you can say, this genetic modification technologies or transgenics research is mother of biofortification research. Then how it has become mother of research, let us see. Then conventional breeding, which has most practical approach. Okay, agronomic approach, if you see, as I told, this is external application of appropriate nutrients depending on the growth stage. So either they are applied in soil, they are applied in foliar, they are given in fertigation. But you see, the cost of application, already farmers are burdened with inputs like fertilizer, pesticides, and other things. So in addition to that, if you ask him to do manual or foliar application or other kinds of applications, external applications, he will not be able to do. And let me also tell you, there is no incentive till now for purchase in the market. See, suppose even if he sprays zinc and he gets high zinc rice, still he will not get higher price in the market because there is no market segregation. So as you can see, this is good as a proof of concept, but it has a lot of biological limitations as well as other practical logistical issues for agronomic approach of biofortification. But as it is successful, suppose you do right time, if you are spraying the panicles leaves with zinc or iron or selenium or iodine, there is definitely enhanced nutrient conditions. So next goes the genetic modification technology. Everybody knows about this transgene technology, introduction of genes from novel sources, from desirable target traits. So as everybody knows, the golden rice, and it is done in all crops. So as Dr. Monica Garg from NABI has put it, it is maximum research but minimum utilized. How? Let us See, one by one. So why transgenics for biofortification? Because there is genetic variability in conventional breeding. Suppose if you see golden rice, vitamin A is not there, or precursors of provitamin A are not there in polished rice. So you have to go to genes outside the system. So novel genes like, and to enhance uptake and transport to the target tissue. As you might be aware, the minerals or nutrients are there in the vegetative mass, which are in the straw or leaf sample. What we need is these minerals should go into the grains, and also to increase the bioavailability. Simultaneous incorporation of many genes that is possible, as I will demonstrate you in my later slides. So you can stack many genes of vitamin A, iron, zinc, if using transgenic technology and targeted expression. They will express only in the your tissue of interest, whether it is grain or cobs or tubers. So transgenics are very advantageous, and there are many genes, especially ferritin, metal chelators, metal transporters, and phytases. And you can see the examples where they have shown successful integration of transgenic development and integration of genes, and leading to the development of high lysine maize and soybeans and cassava and golden rice also. So there are many cases. Transgenics is indeed, from research point of view, is successful technology for the development of biofortified varieties, period. There is no question. But the regulatory issues and other aspects are hindering this transgenics for biofortification. I will just take a few minutes for biofortification in rice. Because rice is ideal crop and also my area of expertise. As you can see, since 1999 they have started developing transgenics for iron, zinc, and their bioavailability using metal chelators and several genes. As you can see, they are from barley, then Phaseolus, there's mung bean, and also from rice. So as you can see, there are, and they have used combinations of genes to express. Actually, if you can see, up to 50 ppm of zinc they have achieved, and about 20 ppm of iron they have achieved in polished rice. And recently, and I am glad to inform the house that, and children should also be, or the students also should be aware that recently golden rice has been released in Philippines on July 23rd from the concerted effects of IRI and Harvest Plus and Golden Rice Consortium. See, as you can see, the research started during 1999, but the variety could be released only in 2021. So it has taken so much time. If you see wheat, so you have transgenics developed from provitamin A, iron bioavailability, protein content, antioxidant content, and also to the quality of the starch of in wheat. So you, as you can see, there are a lot of genes which were targeted, which were transgenics were developed, and proof of concept has been shown. But none of these are not yet released, or they are confined themselves only to the research level. So similar is the case with the maize. You can see provitamin A, because maize is altogether a different story, because it has a lot of commercial aspects. So you can see there are many traits, as I was telling, again vitamin A, vitamin E, vitamin C, multivitamins, and bioavailability, and also protein content. As you can see, several amino acids have been targeted using various techniques of biotechnology, and maize varieties have also been released in Japan, Mexico, and several other countries. So wherever there is commercial aspect is there, even transgenics are accepted. So even the crop has some commercial advantage, the transgenics are accepted and released in those respective countries if their regulatory system is in place for the release of transgenics. Similar is the case with soybean. As you can see, soybean also, because it has commercial implication. So all these vitamin A, cysteine, linoleic acids, all these either they have upregulated the genes of interest or downregulated the genes of interest using transgenic approach. And again, lot of transgenic soybean varieties have been released in many of these countries. Sorghum, but sorghum is not well researched for in terms of transgenics. So they have only few studies for digestibility and provitamin A. So these are some of the transgenics traits of biofortification which are developed across the crops. You can see vegetables and fruits everywhere. All transgenics proof of concept studies are there. You can, even if you just Google it, you will find n number of transgenic papers targeting biofortification of fruits, cereals, vegetables. So let us go to now approaches for conventional breeding. This is much more interesting to us because we can directly use these products without any regulatory constraints, since and we can evaluate the germplasm, we can develop the crossing material and identify the QTLs if they are available, and use mass or MAP for the development of biofortified varieties. And the case of biofortification in terms of conventional breeding is successful in many cereals, potato, sweet potato, vegetables and fruits also. These are some of the varieties which have been released using conventional breeding. So what is the advantage of biofortification? So the capitalizes the large intake of daily intake of food staples regardless of age, gender, economic status. See, rice is rice everywhere. Wheat, chapatis are chapatis everywhere. So we will, our consumption is more. Our consumption is lump sum, and the intake will be enough for micronutrients. Suppose if you are biofortifying rice or wheat, so you are assured that the poorest of poor also can afford these nutrient rich cereals. So one time investment. Suppose I am developing a biofortified variety, it will be there in the system, and it is very sustainable and economic and does not compromise yield per se. We have, everybody knows that we have varietal release system. So there they will see that if they are releasing a variety of biofortified variety, then they will see that the yield is not compromised, and they are, most of the time they are not toxic because they are within the biological system. So as you can see, the proof of concept, everybody knows the QPM maize, and Harvest Plus has funded the biofortification research across the countries, especially in countries of low income and middle income groups, and especially in India, it has provided lot of infrastructure, breeding material, DBT also supported, ICR also supported, and we have lot of biofortified varieties. Any one of you is interested, you can check in ICR website where we have a bulletin where they have shown 70 biofortified crop varieties across the ICR and agricultural system, agricultural universities and National Agriculture Research System. So how we develop biofortified varieties using conventional breeding? So the donors should be available for high nutrient content, because unless there are donors we cannot develop. For rice it is iron and zinc, and as I was telling, vitamin A is not available. However, golden rice is one of the target biofortified crop, and wheat, maize, and you should know that the discovery of maize mutant Uptake 2 led to the development of QPM, and we have markers also for that. Similarly, the natural variation of beta carotene hydroxylase is also now source of development of provitamin rich maize, and pearl millet, iron and zinc. And we should also see about anti-nutritional factors, and we should not restrict ourselves for the donors. We can go for wild species or different genomes or land races. So the donor germplasm is very critical for the targeted trait. So, and as I was telling you, the farmer will not take the biofortified varieties unless it has high yield and desirable quality. So we should see that you are converging three of these: yield, high nutrients, as well as quality. So the conscious breeding efforts are needed. In addition to that, we also need high throughput estimation systems like XRF is needed to identify the promising lines, and influence and soil and environment is much especially if the soil, if the nutrient is being taken from the soil. So this is XRF. This has been provided and it is there even in TNAU. If I remember, we have a couple of XRFs. Dr. John Joel and others, they have this XRF for the analysis of various nutrients. It does a lot of samples. We ourselves, we have done around 1 lakh samples till now. So now let me go to mapping, because mapping is easy in the sense, if you have any major QTL, it is easy to develop the varieties by introgressing the major QTL into the varieties. The idea is this, but why it is not so easy, let me tell you. If suppose you have this nutrient trait is controlled by several genes or genomic regions which are known as QTLs. I think everybody knows about QTLs. So if you see how it is being controlled. So these are some transporters. So like this. So many genes are needed to consortially work for expression of these genes. Suppose if it is controlled by a single gene, like any biotic stress gene, then it is very simple. You transfer that gene, you will get the high zinc. But it is not like that. It is since this biofortification traits are controlled by quantitative traits loci, the issues are complicated. Means for transfer and or for introgressing. So these are some of the, as I was telling, these are the, we should get these. Instead of this, we will end up in getting only these QTLs when we are developing any mapping populations, because we need to pyramid many QTLs to get 100% biofortified targeted trait. So these are some of the QTLs which have been reported in rice. If you can see, there are so many QTLs from so many donors. So which QTL we should use? We should suppose your let us say in Tamil Nadu there is a favorite white pony variety is there. You want to biofortify it, so you should use the same donors under which these QTLs are reported. We cannot take from any QTLs. We should take only that donor in which QTL has been identified. So this is where the conventional breeding is slightly tricky, but still it can be done. So how, once you have developed the variety, so ECRIPS, All India Coordinated Program, has a special trials in rice, in wheat, in maize, and in pearl millet. They have already fixed the levels for optimum iron and zinc. See, they will not release a variety unless there is optimum iron content and optimum zinc content. Similar idea is also mooted for rice, wheat and other crops, so that the only biofortified varieties are varieties with optimum nutrient contents are released. So I will just brush through quickly the biofortified varieties developed. We have around 10 biofortified varieties released for zinc and protein, which have yield high zinc and quality. And similarly, we have 20 varieties of wheat varieties developed and released through central varietal system with either iron, zinc or protein. And we have around more than 15 to 20 hybrids in maize, because it is a different ball game, because they have QPM. Even QPM is considered as biofortified varieties. So with the provitamin A, with the lysine, with the tryptophan, the iron it is difficult. With the zinc also they could find, and as you can see, many hybrids are released. And when they have fed this maize to the poultry, they found that it is increasing the breast muscles in poultry feeding experiments. So since maize is used as a poultry feed mostly, so it also helps the poultry to be healthy. So pearl millet, as I said, optimum amount of nutrients, iron more than 42 ppm, zinc more than 32 ppm are must. Unless you do that, they are not released. As you can see, 9 biofortified varieties have been released in pearl millet, and clinical trial has shown that the iron status of around 250 children have been enhanced when compared with the control. control for millet. Sorghum is already biofortified, but sorghum problem is they are still trying to break that optimum yield, and they are looking for donors for removing, for enhancing the nutrients as well as to remove the anti-nutrients. So outside India also, as you can see, Bangladesh has a lot of varieties. For high zinc rice, they have around 5 to 6 varieties. They also have hybrids. Philippines has one zinc rich variety. Wheat varieties are there in India and Pakistan, which are in pipeline. And if you can see, pro-vitamin A rich maize was released in many African countries. Cassava is also released in African countries. Sweet potato is also released in African countries. And since the African countries, their staple is beans, so they are also looking for iron-rich beans for biofortification. So these are some of, I'll just brush through. These are some of the varieties which are developed in finger millet, so little millet, and lentils. We have pulses also have been biofortified, groundnut, linseed, then mustard. As you can see, we have 00 mustard which is low in erucic acid, and we have soybean. These are from India, and we have cauliflower, we have potato, sweet potato, and yam. Pomegranate also, which our Honorable DG always mentions, this Solapur lal, which is very much rich in biofortified varieties, biofortified fruits, in several minerals. But idea here, pomegranate, if a person can buy pomegranate, he can as well as buy the nutrients. So he can as well as buy some leafy vegetables. So the idea here of biofortified from research is to majorly what you can say strengthen the cereals, because that is the staple food for the poorest of the poor with the low purchase power. And we have collaboration with NIN. They have clearly shown that biofortified cereals or food has advantage over the conventional varieties, just because there is more content of nutrient. So more content of nutrient is also increasing the bioavailability. So we have one consortia research project, CRP and biofortification. As you can see, TNAU is part for rice as well as for maize, and Dr. John Joel, Dr. Jay Prakash, and Dr. D. Sudhakar are active participants in this project, and we are very proud of this program. There are going to be many more varieties in future to come from this platform. And so there is one question is always asked, since we are increasing the biofortified nutrients, are they toxic to us? So as you can, I'll take simple example I will give. If we consume 25 grams of polished rice with 12 ppm, so it gives only 3 ppm and catering only 20%. So, and if you are eating rice with 24 ppm, still it gives only 6 ppm. So that is only 50% of RDA. So the idea is we will not go to severe malnutrition in terms of micronutrients. Otherwise, there is no the question of toxicity doesn't arise from these biofortified varieties, since they are developed within the system. Means we are not injecting or fortifying them. So it is plant is taking, so there is a buffer within the plant. So, and we should mention about the phytic acid, because this is an anti-nutrient. This will bind to the nutrients and it makes it bioavailable, but it is not same. There is a lot of variability in phytic acid also. With we breeders, we should be able to break the linkage between phytic acid, and we should be able to select the recombinants with less phytic acid and more nutrients, and which is quite possible as we are seeing in the breeding programs now. So what are the challenges? So if you see from the developer's point of view, from breeders, combining high yield and high nutrients is slightly difficult. Why slightly difficult? It is a lot of difficult because of the dilution effect and influence of soil and management, micronutrients As if you have time, you can just see the ECRIP data, where the genotypes will vary across the country because of the influence of soil and its the availability of micronutrients, and as I mentioned about anti-nutrients and the complexity of traits of QTLs because of their QTLs. And farmers, why they are interested to grow biofortified rice? Because there is no special price, so no incentive, and lack of awareness. They are not even eating their own crop. So our idea is to create awareness, and so that at least they will, farmers are also equally or with low purchase power. At least they should use this crop with low um, with high nutrient contents, and target consumers. We should promote it as health foods, or whether it should be integrated in PDS as Honorable Prime Minister has indicated. It will be integrated. If not now, it will be definitely integrated in coming days in PDS and midday meal or ICDS. So and let me also tell you, during recent on World Food Day, 17 biofortified crop varieties have been dedicated to a nation by Honorable Prime Minister on World Food Day. This itself shows the importance of biofortified crops as sustainable solution. Thank you very much. I am thankful to the organizers and all the sponsors. If you have any doubts, I am ready to answer.
Thank you very much, madam. This now the topic is open for discussion. The participants, you can interact now with the speaker. The participants online, if you have any questions to be interacted, when they will come out, madam, please.
Not only here, madam, they can always ask me through what phone or email. I'm always available, madam.
Yes. And also you can please in the chat box also. We will forward to
Yeah, I will also check, madam.
Yeah.
Chat box also. I'm not able to open the chat box. Maybe you can tell me if there are any questions in chat box.
Okay.
Maybe it is too fast.
No. No problem. Anyway, we will get back to you, madam. If there are any queries, definitely
Sure.
please to forward to your mail or to your WhatsApp. So coming to the concluding remarks of the session, thank you very much, Dr. C.N. Neeraja, for your nice presentation of the biofortification. And you have highlighted a prelude about malnutrition in India and the ways to combat the malnutrition, especially with the micronutrients, iron and zinc, and also the proteins, and the different activities that you are carrying out in the Directorate of Research and with the different crops, and the various approaches that you are following, including the conventional breeding, and you have very nicely, the speaker has narrated the different crops on which the work is being carried out from the breeder point of view. And we are natural resource management scientists, madam, so our work will focus with the soil application or the foliar application of those nutrients, especially the micronutrients. And you, from the breeding side, you will be concentrating on the breeding aspects. So like that, as you indicated, our TNAU is also a strong partner in this biofortification activities, both in the plant breeding and genetics as well as in the Center for Plant Molecular and Biology. They are also working on the iron fortification and zinc fortification with the rice and other crops, as you mentioned, our Dr. Jaiprakash and John Joel and Dr. Ravindran, Professor in Biotechnology. They are also working on this particular line. Actually, this lecture was very much enlightening to our natural resource management participants, in fact. And in our scientists, we also used to discuss this biofortification from the crop improvement side as well as for the natural resource management scientists, especially from the soil scientists. So we used to interact and we used to come out with many action plan for carrying out this particular research. And also you focused on the phytic acid, the anti-nutritional factor. So it was very informative and emphatic, and the challenges also you were focused. So now, as a concluding remark, we thank you very much on behalf of the organizing committee and the Dean School of Postgraduate Studies, Tamil Nadu Agricultural University. And this is a golden jubilee year for our alma mater, Tamil Nadu Agricultural University. So very kind of you for having joined us. Though there was a little technical problem, with our request, you were able to very neatly focus on your topic within the outcome. Thank you very much, madam. Again, to see you in future endeavors. Thank you.
Thank you, madam. Thank you.
Thank you.
So I'm leaving now, madam. Is it okay?
If you have time, you can join the next speaker. If otherwise, no worry. Please. Thank you.
Thank you.
Yeah. Good afternoon, ma'am.
Good afternoon. I am Dr. Muthu Armugam. Hope you are online. I will request Dr. Vishanti, Professor of Soil Science and Agricultural Chemistry, to introduce the speaker and go on to the speaker. Good afternoon, Director Nurata and the participants of this session. Dr. Muthu Armugam, welcome to this session. To say a few words about Dr. Armugam, he is a senior scientist, microbial processes and technology division in the National Institute for Interdisciplinary Science and Technology, CSIR, Nurata. He has served as a member of the project of DRDO, Army Biodiesel program, and also he has served as a principal investigator of many DSD schemes. Now I invite the speaker to take the floor on the topic bioprospecting of microbial strains for biofuel production. On to the speaker.
Yes. Yeah. Good afternoon, ma'am, and thank you for your kind introductions. Is that my slides are visible and it is in presentation mode?
Yeah, the slides are visible. Please go to the slide view. Slide mode. Yeah.
Is that in
you
is
can
it in
click on
presentation?
yeah, slides are visible. The presentation mode.
Okay.
You can press the F5.
Just 1 second. Ma'am, is it now presentation mode?
Is it visible? Yeah, it is visible now.
And it is in presentation mode also, right?
Yes, it is in the presentation mode.
Okay. Good afternoon to chair and co-chair of the today's theme 4, that is sustainable use of natural resources for food and fuel production. So indeed, I am very much delighted and proud that I am also I am from I am alumni of TNAU. I graduated from Trichy campus. Subsequently, I took my masters in genetics and plant breeding as a CSIR JRF from GKVK, and followed by that, and I joined for my PhD at Indian Institute of Science, Bangalore. Soon after that, I joined in DRDO, and currently I am working as a senior scientist at CSIR NIST Thiruvananthapuram, Kerala. So today's discussion is that bioprospecting of microbial strains for biofuel productions. To begin with, ma'am, my slides are moving. Is that slides are moving, ma'am? Hello? Ma'am, my slides are moving?
Yeah, your slides are visible. You can proceed, please.
Ma'am, is that my slides are moving or not, ma'am? I'm not.
Your slides are moving. The slides are moving. Now you are in the CSIR slide. CSIR.
Okay. I could not put my slides here.
CSIR, National Institute for Interdisciplinary.
Yes, ma'am. Yes, ma'am.
That slide is now on screen.
Yeah. But I could not see my slides here. That is.
Okay.
And just 1 second, ma'am. I'm stopping and sharing again. Is it visible, ma'am, now? Okay. So this is my institute, CSIR. Now it is visible, ma'am. I can go ahead with my presentation.
Yeah,
right?
yeah, it is visible. Yes, it is visible.
Yeah, I have. This is the CSIR, NIST, National Institute for Interdisciplinary Science and Technology. Previously, it was called as a RRL Trivandrum. So for the students' benefits, and this lab is we do have a higher education PhD programs through the ACSR, Academy of Scientific and Innovative Research. ACSR program is there. And to secure this PhD admission, a student need to qualify any national exam from CSIR, UGC or DBT JRF, or from other technical side, GATE exam they need to qualify. If they qualify, we do have a research from chemical sciences and technology, microbial process and technology, from where I am coming from. And we do have a materials group and environmental science and agro sciences and natural product division. Yes. So this is the preview of my presentations. First of all, to know that the energy status, initially what is the energy status, what is the need for an alternative energy, followed by renewable energy sources, and then biofuel, different renewable sources and biofuel initiatives from Government of India, and what are the alternative fuels like microorganism and different methods used to produce those fuel, and fuels which includes biofuel, biodiesel, bioethanol, butanol, and next generation hydrogen and even microbial fuel cells. So far, I will show the work from other groups because I am not directly involved with those researches. And then later, microalgae, I am working with the microalgae as a source for energy and nutraceuticals, more of nutraceutical and a little bit on energy. I will show my own examples, like what are the different strategies we are adapted to enhance the lipid, which is eventually used as a biofuel, and then lipid enhancement strategies, and how selectively to quantify the saturated lipid, because when we go for the biofuel, we need to have more of saturated lipids, short chain saturated fatty acids. That is ideal for the biofuel on the keeping quality and the oxidation status of those. And then the future perspective and discussions. So these are all some few slides. I will show some statistics that India and China these are all the countries which is vastly consuming the fossil fuel. Similarly, here with respect to Indian perspective, you can see that the oil consumption is going at a higher rate whereas the production is almost plateau. We have a limited productions, so that the gap between oil production and consumption is being met from the imports, which incurs a lot of foreign currency and other things. Here is the statistics again, although it's a bit old data, but still it is old good that there is a the need for import is keep on expanding, increasing in an alarming rate. Similarly, the production and other things are coming to the plateau. Therefore, if the government around this world stick to the current policies of fossil fuel utilization, the world will need almost 60% more energy energy than today. What is there being there we are using in 2030, we need 60% more energy. Therefore, keeping this the staggering rate of consumption, the world fossil oil reserve will be exhausted in less than 45 years. As per the IEA report, they predict that it will be lost in less than 45 years. Therefore, we need to have an alternative energy to have an energy security, as well as the to run the industrialization and day-to-day activities. We need to have an alternative source for the energy. So what are all the different forms? These are all, you know that different renewable forms are like geothermal, hydropower, wind and waves and tides. These are all the sources from where we can have the renewable sources. For today's discussion, we are restricted to the transportation fuel. The transportation fuels are like alcohols and biomass-based diesel fuels, and from there biodiesel we can get the esterified fatty acids, which will become the biodiesel, and renewable hydrocarbon diesel. Similarly, other alternate fuel like methane, natural gases, hydrogen, synthetic fuels, DME. These are all the some kind of some of the transportation fuel other than conventional fossil fuel like petrol and diesel. So now I will move into the different sources of renewables like microorganisms. We can use the diverse microorganisms as a directly as a fuel source. For example, here in these organisms, the biomass for fuel product directly the biomass of that organism we are using as a fuel source. For example, microalga, oleaginous microalga, oil-rich microalga, which is rich in neutral lipids, stored. It will store the neutral lipid as an energy reserve. So those things we are making use of it. Similarly, not only microalga, there are other bacteria like bacillus, fungi, and yeast also having the lipid bodies through which it stores the lipid. So those lipids directly we can use as a fossil fuel. And the other one is microorganism as an indirect fuel source. We are making use of the microorganism for conversion of the prime. It helps to convert the certain sources as a indirect fuel source. For example, a lot of methanogenic fermentation is there, ethanolic fermentation by yeast, and fermentation of syngases are there. So which will convert the primary product after the respective methanogenic fermentation or ethanolic fermentation. It will convert it as a fuel sources. In addition, we do have the microbial fuel cells, wherein directly we are capturing the electron flow from the microbial electron transport chain, and that also although it is very limited and it's a concept based things, it may not at present meet out the practical needs, but we can use it this transfer electron transfer from the ETC. We can capture the electron that can be converted as a electricity directly. So we will see the details of these things later in my slide. So now to promote this alternate fuel or to have a country with energy security, the national policy on biofuel being framed by the government of India, and it framed a national biofuel policy on 2018. Later on at 2017, they reviewed with the different targets for blending target of 10% initially they said for the ethanol and 20% biodiesel by 2017, although we could not meet out that one. So during that period, I was in DRDO at that time. DRDO Army Biodiesel Program was mega program. that was started using Jatropha carcass. It's a non-edible oil-bearing plant as a feedstock. Similarly, Camelina sativa and the microalgae. I am taking care of the microalgae, so I will be talking more on the microalgae. So these are the vision and the recommendation of alternative fuels in Government of India, which having the 20% blend with ethanol E20. Similarly, other things, other 10% blend with 3% blend with methanol. All those things, different things, E10, E20, and M3. These are all the government vision on alternate fuel to meet out the energy security and not depending on the other countries for to meet out the transportation fuel requirement, as well as to have a security within our country, energy security. So these are all the DRDO Army Biodiesel program, from where the strain optimization, the success of the program depends on the ideal strain, looking for an ideal strain which has the hyperlipid production things. So those lipids also should be favored with short chain saturated lipids, having more proportion of saturated lipids, which is ideal for the biodiesel program. So this is the tank where the strain improvement will happen, and then large scale and little bit we have demonstrated in 2000 liter scale as well. So microalgae, as I told you in the beginning of my presentations, microalgae having many bio components. Like it is a good source for lipids, which is called oleaginous microalgae, which is we are making use of those oleaginous microalgae towards lipid productions. So these lipids having like high value, low volume product like omega-3 fatty acids. Currently, I am focusing on those as well. And there are lipids which is short chain saturated, like palmitic acid rich organisms are there. Those lipids we can use for biofuel purpose. In addition to lipids, we do have the proteins and the pigments like astaxanthin and all. Those are all the nutraceutically important product which we can get from the alga. So what the potential microalgae, initially ASP from Department of Energy from USA, also aquatic species program they have started, and they made extensive research for 2 decades. Later on, 2 decades, and then they come up with that the microalgae having the potential, theoretical potential. However, it has a lot of technical challenges. If you see these tables, like if you are comparing microalgae with almost 30% oil content, there are algae with the 30%. There are algae like Botryophyllus and all, it has 70% oil by weight. Means if you take 1 kg, in that 700 grams, 700 gram, that is 70% is rich in oil, because the entire oil is stored as a neutral lipid. Other than the structural lipids, there are energy reserve neutral lipids which is stored as an energy reserve. If you have this one, the productivity when compared to the terrestrial plant like corn or coconut or sugarcane, we can meet out so much biodiesel productivity per unit area is possible. Because how it is possible? Because it's fast, it synthesis and accumulate large quantity of neutral lipids. When compared to the terrestrial plant like any oleaginous plant, corn or maize, or any oil bearing plants, here 22 on weight by weight basis, it is most of the proportion is enriched with the neutral lipid. Similarly, it has the highest growth rate, doubling time. Per day, it's almost like nanochloropsis or chloral and all. It is a 3 hours doubling time. So every 3 hours, it's keep on doubling. Therefore, it advances many fold within short span of time. In every 10 or 14 days, we can go for the first round of harvesting. Subsequently, every 3 days or 4th day, we can keep on harvesting. Therefore, we could achieve the more productivity per unit time and unit area. Similarly, it uses the wastewater, nutrient from wastewaters, and the highest advantage is that it sequesters the CO2. It takes the CO2, being in a photosynthetic organism, it utilizes the CO2 for the bioconversion of the lipid and other things. Like the biofuel which we are getting, which is having an advantage that it don't have SARS, NOx, like that, such kind of a toxic thing it won't release. So it is environment friendly. And we can, without any seasonal independent manner, it can be cultured anytime by indoor cultivation as well as the outdoor also by using without interfering with the inputs for agriculture. So considering all these points, algae is superior over the terrestrial crop plants to meet out the energy demand. Different steps are involved in algal biofuel R&D. Like it starts with the species ideal species selection and then cultivation followed by harvesting. Harvesting is the one of the bottleneck, and a lot of technical challenges are required to engineering challenge to make the harvesting, because it's being an aquatic organism. How we are dewatering, harvesting, it needs almost 20 to 30% of the total production cost goes for harvesting. We cannot go with energy intensive centrifugation method, and therefore at present we are using some natural polymer. You can see in this panel it is settling within 5 minutes, it is settling. But this is good for the biofuel application but not for edible other things, because the polymer which is coated over the cells may have some hindrance when we are going for edible application like omega-3 fatty acid or selenoprotein, those kind of things. Biomass and conversions. Similarly, this is the when you have the algal biomass, we can go for different ways. We can go to be specific, direct combustion. We can go, and from there, bio crude will come, and we can generate the electricity that from the direct combustion. Similarly, different route like anaerobic digestion we can use to generate the biogas. After these are all the things after extracting the primary product. For example, I am interested in omega-3 fatty acid. You can extract the omega-3 fatty acid, the de-oiled biomass, the left out biomass, currently being used as a undervalued product. So those product you can convert for that, because this is rich in proteins and other polysaccharides. So we can use that for anaerobic digestion. Similarly, the fermentation to get the ethanol, and even for hydrogen productions through the photobiological hydrogen production conversion. This is one aspect. The other one is we can also go for a thermochemicals, pyrolysis and other things. Depending on the biomass availability, we can go for at high temperature and pressure, we can go for thermochemical conversion to get different products. So the alternate fuels from algal biomass, not one is the biodiesel. After getting that is comes from the lipids. Generally, the lipid whatever we are getting in terms of triglyceride needs to be trans-esterified with appropriate catalyst, both alkali as well as acid catalyst. This will be converted into the respective methyl ester. This methyl ester only directly we can use for running the engine, because this is compatible for the existing engine without any modifications. Similarly, other than biodiesel, we also can go with the biogas and methane, as I explained before. Similarly, bioethanol and the butanol we can get here. This is depending on the biomass, the protein and carbohydrate rich biomass is their algal biomass. By using the appropriate organism, we can go for the plural. This is the starch content. By using the appropriate microorganism, we can go for specific conversion of acetone, butanol, or ethanol, depending on the, for example, Clostridium. If we have, we can Clostridium acetobacterium. By using the Clostridium, we can go for butanol production. Similarly, different organism, by using the different organisms, we can go for specific fuels. So after getting the biodiesel, whatever the sources from microalga or from yeast or from bacteria, there are some specific standard specifications are there to meet out the B100 means 100% biodiesel, or with the blend it should meet out with our Indian standard or American standard or European standard norms needs to be met out, which is nothing but it has the calorific value, but different iodine number, ignition value. Those are all some specified numbers are there. It should meet out those numbers. So far we have seen that different way of different alternate fuels. The other one is the currently being even the country today's policy. They are going to come up with a big project on R&D for bio hydrogen productions. Hydrogen means by using the direct photolysis, we can get the by photolysis of water in the presence of light, we can get hydrogen and oxygens. So this hydrogen directly can be captured. And the limitation here is that storing the, all of you that hydrogen is a highly labile, means thermo unstable. So therefore it needs to be properly stored, otherwise some fire related hazards will come. How to store the things which can be converted and used for a moving transportation fuel, that is a big challenge. The engineering disciplines, people from engineering side, they are working on it. The other one is indirect photolysis, wherein initially we are converting like it involves two steps. You can see here that during the photosynthesis, the water molecules are converted by microalgae into hydrogen ions and oxygen. Later on, this hydrogen ion are converted into hydrogen by using the hydrogenase enzyme, which can be converted. This hydrogenase enzyme is highly, it will be rapidly inhibited by the oxygen, which is produced as a co-product from the photolysis. So therefore, it must be produced in anaerobic conditions. Otherwise, the hydrogenase enzyme like this rhizobium also having the root nodules to protect from the anaerobic, to protect from the rapid inhibition of oxygen, it protected through root nodules. Similar manner, these hydrogenase enzymes also need to be protected. In order to have that, it can be done in a two different thing. In first step, in a natural conditions, the first stage, algae is grown in a photosynthetically under normal condition. Later on, when it grows, later on it will be moved, the change mode of nutrition is changed, medium is changed, wherein the sulfur is deprived from the media. Once sulfur is deprived in the media, the microalgae are fundamentally altering its photosynthesis as well as its cellular metabolism to survive. So at that time, it consumes the internal starch and protein and produce the hydrogen. In this manner, the theoretical maximum hydrogen production was reported to be 20 gram hydrogen per meter square per day, which is, although it is a bit old statistics, but this is the one of the landmark thing people have established it. And this is one of the upcoming thing wherein to produce the maximum amount of hydrogen and also storage, if we are using our knowledge or the scope is there in future, specific themes funded by the government of India, a lot of projects will come, specific calls will come on biohydrogens. So if anyone of us is interested or having expertise in this line, we can think of those areas. Other one is continuous mode of hydrogen generations. In this mode, the electrons and protons that are released during the photosynthetic hydrogen photosynthetic oxidation of water are directly recombined by the hydrogenase to produce the hydrogen. This is yet another compartment is there to continuously produce, continuously harvest the hydrogen, store the hydrogen produced during the oxidation of water. In this manner, it is having 33% more efficient than the previous one. And also, a lot of technical and physiological parameters of microalgae cultivation is influenced for the efficient production of hydrogen. And by optimizing those parameters, we can have the more amount of hydrogen generation. Other one is thermochemical conversion. It is like under high temperature and atmosphere, high pressure, we are converting the biomass for the bio crude, which having the specific calorific value. Microbial fuel cells. Here, due to the running short of time, I will just rush these things. Here, the electron and proton, which is it consists of two chambers. One is axic and the other one is anaxic chamber, which is divided by the membrane. You can see here, there is a membrane here. And when the biomass is delivered, the bacteria start growing. when it is growing, the active electron transport is happening. At that time, the proton will pass through. The whatever the electron in ETC is passing through, that will be captured for the electricity generations. So far, we have seen that the work. What is the need for an alternative fuel or the biofuel, and what are the different biofuel options are there, and how we can use it, including the microbial fuel cells. Briefly, we have seen these are all the work from other groups. So now onwards, I will in next 10 minutes I will share you the work which I am doing currently in my lab. Different approaches are for the student. After knowing the different biofuels, the student from masters and PhD level students, for them to have some kind of motivation towards research, you can go through that. There are different approaches are there to produce the biofuel, desired biofuel, any biofuel of our interest. So those things should have, first of all, the strain stability should be there when we are scaling up in higher scale, or when it is producing, there are feedback inhibition kind of thing. So when we are the strain should be stable in a proper condition. That is the asset of a researcher which he is developing. Whatever the strain he is getting, it should be cope up with the scalability. Scalable means whatever we are demonstrated in the lab level should have the scalable when it is going to higher different kiloliter level. Different reactor mode, it should go per scale. It should be scalable, and it should have the highest productivity. So you can see here there are classical microbiology methods, the cultural optimization like growth parameter, nutrient. I do have the nitrogenal stress starvation. We will see a couple of slides from there. The fermentation, classical fermentation method. Similarly, the molecular and recombinant DNA based approach, wherein the classical genome editing, CRISPR-Cas9, all those things we can use to enhance the product of our interest by eliminating all other pathways. Metabolomic approach, using system biology and metabolomic approach. So these are all the different approaches. All these methods in a given 45 minutes we cannot talk. It will become more superficial and diffused. So therefore, I will take my own example, certain things, what is being done. I will give a gist if you can understand, and it will be very useful. And later on, we can have a discussion. For example, the metabolomic engineering, wherein it is metabolomic engineering involves the optimization of a genetic and regulatory process within the cells to increase the production of certain substances. Let it be for other therapeutic substances or a biofuel interest, we can enhance. For example, here you can see that this is the geranyl pyrophosphate pathway, wherein the lutein is the, suppose I need a lutein, whatever the synthesis is the common substrate, trans-lycopene is the common precursor for lutein as well as the neoxanthin. Because when it goes to bifurcated into two pathways, the desired yield of our product will be reduced. So I will block this trans-lycopene being converted to neoxanthin. I will take, so this is the metabolically engineered strain which produces the lutein in more amount. So in this manner only we can have the strain of our choice, which is good for producing certain product or biofuel of our interest, which will be produced. So in this manner, we have very recently in 2020, we published a paper wherein the metabolomic, instead of global metabolites, we are targeted to certain metabolite like GABA, and when we give the nitrogen stress, we are getting the more amount of triacylglycerol. This is lipid bodies. During such times, we have chosen, for example, here you can see that these are all the targeted metabolites like GABA, glutamate, amino acid glutamate. Similarly, the other cofactor like NAD, NADPH. So these are all the factors which is having central metabolism, role in central metabolism. TCA cycle is very important because acetyl-CoA is a precursor for lipid synthesis. So therefore, by enhancing this component, which this will go for other things as well, but by enriching these things, we have done the LCMS, this metabolite, its level during the nitrogen starvation we gave. Nitrogen starvation, we have shown that it having more amount of lipid. At that time, what are these targeted metabolites? What is its level? For example, you can see NADH. which is going beyond the time. It is going with the time. So when we are increasing the time, it needs its levels are going high. So if you are providing more, it's like positive correlations. When you are providing these metabolite, we can have the enhanced amount of lipid productions. Those lipid in turn can be used as a either nutraceutical if it is a long chain fatty acids. If it is a short chain saturated fatty acid, we can go for the lipids depending on the need. So what is the high value, low volume products are more commercially sustainable. In another similar manner, when we gave the plant growth regulators, which triggered the metabolomic profile, which in turn leads to the enhanced lipid accumulations. So this is recently we published in Journal of Applied Phycology. Yeah. In addition, there are many other strategies. Every time we need not go with the gene and CRISPR-Cas for genome editing. By adapting simple culture strategy methods also, we can get the more amount of lipid. For example, here we used the nutrient stress and nitrogen starvation primarily. In this cartoon, you can see that there is a lipid in normal conditions. When we are providing the nitrogen nutritional stress, it leads to increased amount of lipid. So during that time, what happens, including the metabolomic, other things also we questioned, like these are all the lipid bodies. Using a lipophilic dye, we can quantify, and the lipid body can be seen by SEM. And then we do have GCMS, so we can profile it, what kind of fatty acid is being there. If it is short chain saturated fatty acid, we can go for the biofuel purpose. If it is a long chain fatty acids, it is more useful to go with the nutraceutical applications. Similarly, during that nitrogen stress, we know that when we give the stress, it shows the more amount of lipid. But what are the genes responsible, or what are the molecular event happening during that time was largely unknown. So therefore, we have taken the during that stress condition, what are the differentially expressed genes and in turn protein products. When we went with the initial single dimension SDS page, we could not make out, although there is a difference. So therefore, we went with 2D, the proteomic tool, 2D, two-dimensional SDS page. You can see that certain things, for example, this stress associated SAP we are calling, the SAP1, which is exclusively present, which is completely absent in the nitrogen rich condition, whereas in the nitrogen starved condition, you can see that it is exclusively only present during that. Similarly, the SAP7. So these are all the protein we have identified through MALDI-TAB, and we quantified also relative level of expression. And we found that it is a LALDAC endonucleases, which is of mitochondrial origins. The experiments are in progress to reach to certain level that what it is doing during the nitrogen starvations. Does it has any relevance with the lipid synthesis or playing with any other network crosstalk between other metabolic pathway? We are analyzing those. Yes, and I will take another just 5 more minutes. I am come to an end of the things. So when we are the bioprospecting of microbial sources, any, let it be a bacteria, yeast, or microalga, if you are having many hundreds of organisms or under different treatments, what is important is rapidly how accurately we can quantify the level of saturation of the fatty acids. As I told you that if it is a saturated, it has some other utility. If it is a polyunsaturated fatty acid, which has specific functions. To do that, we have used the SERS, surface enhanced Raman scattering. This is the technique using the Raman spectroscopy we can use. And from there, these are all the conditions. For the want of time, I just skip this one. And using this is a Raman substrate to enhance the signal we have used, that is the gold nanoparticles. These are Raman things. So, yeah, here you can see that based on this pattern, the spectra we are getting from Raman spectra, if the fatty acid peak comes at 1622, it is mostly related with the alkyl cis structures, which indicates more of unsaturation indicator. Similarly, for saturations, there are some characteristic peak we have identified, which comes at a specific peak length when we are going the long range reading. like the peaks coming from 1542 and 1444 spike, whenever it comes, it indicates the saturations. So based on certain few three markers like that, very acutely, rapidly, in a large number of sample can be within a less number of time, or in a single go, we can make out which is saturated, which is unsaturated very rapidly. If you want, in other way, classically, we have to isolate the lipid and transestify. If you want to read through GCMS, we have to transestify. It will tell individually, like C16 is this much, C18 is this much, like that. It will individually tell. It is time consuming, although it is very precise. Here we can qualitatively, very quickly, we can come to know that whether it is a saturated or unsaturated lipid based on this marker. So I think almost I come to an end of the impact of the studies. As I told you that very quickly we can go, come to a conclusion that these are all the strains which is good for edible nutraceutical applications. These are all the strains which is good for the biofuel application, because which is having more of saturated fatty acids. So this is the impact which is find this study find it's a better place, a decent place in its thing. So with this, I think I have completed in time, and I thank all the funding people and my student and collaborators. If you students having any specific questions, because through online mode we cannot, we don't have an eye contact. I cannot convey the things properly and explain the things. If anyone interested having any specific questions now or future, you can always contact me. Thank you, and I'm happy to take up any questions if available.
For discussion, any questions from the students as well as from the audience?
Hello, sir. I'm Dr. Kalechel, professor of microbiology. We are also working on other biofuels for past 7 or 10 years. So actually, we have worked some cultures with good quality, high quality, and around 40% lipid content of Aptodismus, Chlamydomonas, Chlorella, and our those species from daily biosphere. And we have also studied about we are also studying about the strategies for enhancing lipid accumulation. Like we are doing it with the chlorella, and we have done gamma mutation also. The wild is fixing around 28% liquid. It accumulates around 28%, and one of the mutants is accumulating 40%. Now we are in a state of characterizing that using RAPD and then 2D in order to locate which genetic is responsible for enhancing this particular for accumulation of more of this fetal lipid. So that kind of work we are doing it now. We have started in with aquatic species program, and we have experimented with many strategies. So still, it is still in a laboratory stage. What would be the reason for it? Why it is not come to the field level? How to?
Yes, ma'am. Actually, I'm not clearly getting your side audio is not coming properly. I could not get properly. We can your question is not clear to me, ma'am.
Actually, it was all started with the ASP, Aquatic Species Program during 1970s by USDA people, and they were stopped in between after 90s. Again, they were started, and global level, even in India, we are also working with many algal species. Still, it has not come. It has not come to the field level. It is still in laboratory conditions. So, what are the strategies? What are the ways you are suggesting for bringing it to fields again?
Yes, ma'am. Actually, through ASP program, which is started as a part of DOE funded project in USA in University of Colorado, they conducted for 2 decades, and they completed the study with the 2 conclusion. That is, algae is having theoretical potential. However, it has a lot of technical challenges. So therefore, they have concluded it. But later, we now only we have started up. That is why instead of algal oil being converted to biofuel side is it is now it is not sustainable for biofuel. Rather, same amount of lipids if you are converting as a omega-3 fatty acids like EPA, which I am concentrating, but that is the low volume and high value product. We cannot compete with the fossil fuel like petrol, diesel. In that manner, to that rate, we cannot give the fat lipids we are extracted from the algae. After converting, we cannot compete with the current petrol, diesel. So therefore, it is economically it is not feasible. However, we can make use of this.
Can we go for field scale production using biodiesel?
We can scale up, and even for some other reason through my DBT project, we demonstrated at 1,500 liter scale in commercial farm in open raceway pond or production of EPA, that we can scale it up. Scaling up is not a problem, but when you are scaling up, the energy efficient harvesting method should be there. That is another limitation.
Yes. Okay, thank you. Thank you so much. Thank you.
Yes.
So, to conclude with, we thank Dr. Nurata for his explicit talk on the microbial role on the biofuels, especially the algal ones. So, microalgae indeed is playing a great role in the renewable energy source. So, it is a great insight into our researchers also. We are happy that our own student is doing an excellent work in the DRDO on this aspect. And we are a bit sorry that we cannot spare much time for a very extensive talk and for a very good interaction session. However, we thank you, Dr. Aruna, for your beautiful, excellent work, portraying your work also. Thank you so much for sensitizing us.
So much. Yeah. So now I think with the permission of the chair, I will leave the session so the student can go ahead with their presentations.
Okay, very good. Thank you very much.
Thank you, ma'am. Thank you so much.
So now coming to the next session, the presentation, oral presentation by the student participants. There are about 5 presentations. Ms. Pavitra, Navina, Kadipin, Nishala, and Rovia. And for the information of the participants, there is a oral presentation of which content of the presentation, clarity and flow of the presentation, novelty or contribution to the field, ability to interact and or discuss, and timing. So these were taken into account for evaluating your presentation. And of the 5, 1 will be awarded with the Best Oral Presentation Award. So this is for the information of all the participants. And now, one request to all the participants is the time allotted is 10 minutes. So please stick on to the time, of which 8 minutes you do your presentation and about 2 minutes we will go with the interaction. So now first I call upon Ms. Pavitra, you please share your slides and start your presentation. Please keep up the timings.
Okay, ma'am. Thank you, ma'am. Good afternoon, one and all. I hope. Okay, ma'am. Good afternoon, one and all. I hope my slides are visible. Good afternoon, DNRM. Professors belong to the department under the DNRM. Myself, Pavitra. I am second PhD scholar from the Department of Soil Science. Next slide, please. As you have seen, the topic is all about the delineating the fluoride contamination and relationship with other chemical properties of the soil. So we have the preliminary. Hello, ma'am. There is some.
Pavitra, you can proceed, Pavitra.
Okay, ma'am. I hear some voices, that's why. As you have seen, the topic is all about delineating the fluoride contamination and its relationship with other chemical properties of the soil. The study area is Rediyachatiram block of the Dindigul district. We have gone for survey in all 23 villages for the block, collecting the soil and water samples. Next slide. The you have know that you all know that the elevated fluoride concentration. Is there any elevated fluoride concentration in soil and water became a major problem in 25 countries that affects the 200 billion people. So, the the analyzing the soil property is a major task to delineating the fluoride contamination and correlating with the other soil. As you have know that the little amount of fluoride is essential and therefore that is toxic. That's why commonly referred as a two-edged knife. F contamination is majorly due to both natural and the anthropic resources. The main natural resources are the rocks such as apatite, fluoroapatite, and the anthropic resources are aluminum industries using of the phosphorus fertilizers such as the, the limit of the soil F is up to 6 to 16 ppm based on the soil type and the amount with the available form where the total F is not calculated. So we have gone for the survey. The villages, 80 samples are collected from the according to the standard procedure. The analysis of soil just such as pH, EC, ED, NPK, also organic matter, calcium, magnesium, sodium, and water soluble according to the standard protocol. We have the feel the what fluoride is a toxic, other forms is not that of importance. So we have analyzed only a water soluble fluoride using a selective electrode with the aid of and the results analyzed results of the fluoride are correlated with the parameter. Next slide. And the result of Chatiram is having a neutral to alkaline reaction, moderate conductivity status in organic matter. To the overall block, the fluoride contamination is varied from 0.98 ppm to 3.3 ppm. Stable soil. Next slide. Next slide. Among the we have correlated the water soluble fluoride with other soil properties. This is a correlation table that shows how the pH range, pH and the other soil properties range are. What is the mean value of the pH, pH of that bag, and what is the mean range of the fluoride in that bag, and what its correlation with the other properties. The study shows a pH equal to alkyl reaction with the mean 8.27 and the water soluble fluoride is significant correlated with the that should be R value of 0.31. This implies that is the major consequences of fluoride contamination. And other apart from that factors, the available phosphorus is also shown negatively significant correlated with fluoride. As you have shown, minus 0.213. This means that the available fluoride concentration has something to do with that F property. So, as per previous studies shown that the phosphorus also plays a dominant role in fluoride solubility. Thus, the concentration of the soluble process, insoluble process is increased when the fluoride concentration is decreased. Next slide. So this is a correlation graph calculated by using analyzed parameter. Here shows that the organic carbon is also shows a negative correlation with the water soluble fluoride. What indicates that the organic carbon seizes the mobility of the fluoride into the soil and arrests the fluoride transformation from the soil to the plant. Thus, thus significantly decreasing the bioaccumulation of the fluoride crops, which all the way causes the human food chain. Thus, we have to reduce the bioaccumulation. So, organic matter plays an important role in the immobilization of the fluoride. A positive correlation is shown between the soil pH and the fluoride content, which also shows that the pH, with the increase of the pH, the fluoride concentration also increases. And the correlations of the fluoride and soil phosphorus is negative. That shows that the water soluble fluoride showing a significantly negative correlation, indicating that the F concentration leads to the formation of the insoluble phosphorus compound. I have mentioned that amount of the fluoride, for example, in soil having a more soil but in the haven't used the phosphorus as compared to the phosphorus acid soil. If rock phosphorus is majorly used in the tea having acidic, the fluoride contamination is high because rock phosphorus containing the significant amount of the fluoride and type of the there are some crops which are a good accumulator of the fluoride, such as bendy and Bendy and tomato, where others are not, such as onion and brinjal, and having a physical and the chemical properties of the soil. Soil with having a good texture, low fluoride concentration, the soil with the high clay nature showing high fluoride concentration. Also, chemical properties such as TNAU plays a prominent role in a fluoride contamination. Also, soil pH having the profound effect of the biogeochemistry of the fluoride. The soil pH moderate to alkaline, fluoride contamination is uh, the fluoride contamination is thus in the way it automatically bioaccumulates into the plant, forming the biogeochemistry cycle of the fluoride, which goes into the fluorine gas in the atmosphere that automatically fixes in the soil, so that the biogeochemistry is continued. So soil pH having a major profound effect in a biogeochemistry of the fluoride. High organic matter content increases the fluoride absorption capacity because organic matter having a some function group which was profoundly absorbing the fluoride and controls the immobilization. F showing the negative correlation with ECTS, organic carbon, and the available calcium. Uh, thus giving the idea that the increasing organic matter and the calcium causes the immobilization of the fluoride. That means calcium uh, can complexes the fluoride as a calcium fluoride, thus ceasing the mobility of the fluoride into the soil. Thereby, we are reducing the bioaccumulation of the fluoride in the soil. Next slide. Okay, this is the slide that explains the fluoride toxicity. As I have said, fluoride is growing in an alarming rate in all 25 countries, which affect the 300 billion people. To this biogeochemistry of fluoride, oil, plant, atmosphere contamination. Acute fluoride is majorly due to some industrial toxicity. But we are talking about only a chronic fluoride toxicity. In 2018, World Health Organization alarms that the drinking water of the 300 billion people having a fluoride concentration more than 1.30 ppm, where the recommended is 1.1 ppm in water, and just showing that the more than 300 people in the world are drinking a water with more than 1.50 milligram per liter of fluoride in water. Some amount of the fluoride for adjoining muscles and dental problems. About that the concentration crosses dental fluorosis and the osteofluorosis. That is what the slide explains. I hope my presentation explains what the fluoride toxicity.
Pavitra. Vikram, are you hearing us?
Ma'am,
Vikram, are you hearing us? Vikram?
yes, ma'am, am I audible?
Now you are audible. Over, presentation over.
Yes, ma'am.
Okay. Is there any amendment to come over this problem? The
Yes,
effect
ma'am.
of food
We have conducted the field study. Following for this study, that same blood having a F concentration over a 3.20 ppm, ma'am, somewhere between Silvarpatti village, ma'am. We have given a different amount of amendments such as lime, gypsum, vermicompost, FOM to combined with the SPGS recommended NPK fertilizers and analyze what are the liking for F accumulation. We have find that
Okay, is there any questions from other participants?
the FOM and vermicompost, ma'am.
Is there any more questions from other participants? Okay, if there are no more questions, let us move on to the next presenter. Miss Navina. Navina, are you hearing us? Navina? Navina?
I can hear, ma'am.
Okay. So you can start your presentation. I think you have listened to the instruction, no? You are allotted with the 10 minutes totally. Okay. There is an evaluation. Please start.
Good evening to one and all, Professor here. I myself Navina from I'm doing PG in AC and Arai curriculum campus. I'm here to present about the topic effect of nitrogen sources, micronutrients, and the humic acid on growth and yield of paddy in an alpha of Thamarabarani command area. Next slide, please. So here we know that rice is a stable food of more than 60% of the world population. Every year we need to increase the rice production by 1% to meet the growing demand of food. So in order to achieve these targets, sustainability in attaining a higher yield and intensification of rice cultivations are needed. So as we know that nitrogen fertilizers plays a major role in higher rice grain production. Likewise, in this study, two types of nitrogen fertilizers were used. One is urea and another one is ANA solution, that is ammonium nitrate and ammonium sulfate solution. These ANA solutions are collected from zirconium. It's a byproduct of zirconium, and urea contains a 46% of nitrogen and ANAS contains 23% of nitrogen. And in addition to that, humic acid, liquid micronutrients and water soluble fertilizers are also used. That humic acid which acts as a photosensitize, and also it stimulates the rice growth and productivity. So the main objective of this study is to investigate the nitrogen source, humic acid, and the foliar spray of liquid micronutrients on growth yield of rice crop, and nutrient availability in soil, and crop uptake and economic of different nutrient management practices of rice. So these experiments were carried out in AC and RI curriculum, and it's laid out in a randomized block design with the ASD 16 rice variety. So there are 11 treatment combined. There are 11 treatment combinations, and it were replicated thrice. So the experimental plots were applied with STC or IPNS recommended dose of NPK, that is 110 60 kg per hectare as per the fertilizer schedule. And foliar spray of liquid micronutrient, humic acid, and water soluble fertilizers was sprayed at the time of 30, 45, and 60 days after transplanting. And soil application of humic acid at 2 and 4 kg per hectare at 4 different stages. So these data depicted a significant influence of soil application and foliar spray of various fertilizers on plant height and number of tillers. here we can see that T9 and T4 treatment shows are on par with each other. It shows the highest plant height and the number of productive tillers. So the higher plant height are due to the foliar application of nutrients for proper crop nutrition and optimum growth, and as well as these increases the meristematic cell activity and cell elongation, and these nutrients are known to be beneficial effects on metabolic process, and it improves the vegetative growth. So, and because of the increased photosynthetic rate, the young seedling had better root growth development, and cell division, cell enlargement, and these are resulting in the highest plant height and the number of tillers per meter square. So, and also the higher tillering occurs as a result of improved root growth and increased nitrogen mobility in a soil solution and absorption by plants. Next slide, please. So here that significant increase in 1000 grain weight, grain yield, and straw yield. Here these are the highest grain yield, straw yield, and 1000 grain weight are noted in the T9 treatment and T4. Mm, and the T9 and the T4 are on par with each other, which was significantly superior over all other the treatments. So here that 24.65 gram of 1000 grain weight and 7038 kg per hectare of grain yield and 9115 kg per hectare of straw yield were noted in T9. So these are due to the attributed to increase in supply of photosynthesis to zinc as a result of higher chlorophyll and photosynthesis due to sufficient micronutrients and available, and these micronutrients are available through foliar sprays at uh, uh, various intervals of crop growing period. So, and next, the better nutrition, plant growth, and nutrient uptake are favored in the yield components. Next slide, please. So next slide, please. Next, they-
லேட்டா ரிசீவ் ஆகுதுன்னு நினைக்கிறேன். அவங்க நெட்வொர்க் இஷ்யூனா லேட்டா ரிசீவ்
so
ஆகுது.
nutrient here this table shows the nutrient availability. Here the significant improvement in available NPK was evidenced with the conjoint application of humic acid, micronutrients, and water soluble fertilizers. Among these treatments, T9 and T4 are registered a higher availability due to the increase in the nutrients maybe due to the effect of organic acids produced during the process of decomposition of humic acid, so which might enhances the solubility of native nutrients, and the available P will be increased also due to mineralization of soil organic phosphorus, and available potassium also increased due to the enhancement of microbial activity due to humic acid foliar applications, um, through reducing its fixation in soil and the dissolution of fixed decay. Next slide, please. So here the nutrient content is present in rice straw. Here that T9 and T11 treatment on par with each other, which receives the foliar application of humic acid, water soluble fertilizer, and 2% of liquid micronutrients. Here that the humic acid foliar spray had impact on the protein and the nucleic acid synthesis, which indirectly indicates the increased nutrient content of N and nitrogen and potassium elements. And humic acid also maintains the high level of acidic phosphatase activity. So thus increases the phosphatase activity that holds key for increases the phosphorus uptake by plants. So next slide, please. So here that it shows the micronutrient uptake. If the significant increases in micronutrient uptake could be ascribed to the presence of chelated micronutrients in humic substances, So that and a foliar spray of liquid micronutrients and the humic acids are well known as a complexing agents for transition metal cations. So thereby it facilitates the enhanced micronutrient uptake in crops. And that the foliar application of humic acid and micronutrient enhances the nutrient uptake in rice crop. So here that which receives the liquid micronutrient as a foliar spray in a treatment that shows the highest micronutrient content. Next slide, please. So here that agronomic nitrogen use efficiency and BC ratios are depicted in this table. Here that the nutrient management practices help in enhancing the nutrient recovered by crops and maximizing the yield and minimizing the loss of nutrients, and it leads to the enhanced nutrient use efficiency. Here T9 treatment shows that maximum agronomic use efficiency, and it's followed by that T4 treatment. Mm, in coming to that BC ratio, it indicates that gross return in rupees per hectare were worked out from the yield of rice by prevailing market price at the time of harvest, and it was explicated that maximum net return registered in the treatment of T9, and it indicated that net return decreases with a decrease in availability of nutrients. So the highest BC ratio 2.85 were observed in the T9 treatment. Coming to the conclusion, so these experimental results shows that STCR IPNS NS urea and combined with the foliar spray of 1% humic acid and 2% of water soluble fertilizers and 2% of liquid micronutrients gave the highest plant height and the highest number of productive tillers, and the grain yield is dry yield. So these are due to the effective utilization of well applied fertilizers in addition to the so-called foliar spray of all macro and micronutrients. And also humic acid also performed favorably in the improvement in the yield of rice. So and the yield of rice was 15.6% higher than where the SPCR IPNS N application of urea alone, and this above treatment is followed by STCR IPNS NS urea and along with the 4 kg of humic acid soil application. It shows the 12.24% increase in rice yield than the conventional practice. So next slide, please. So that then the NPK content were also increased in that STCR IPNS as urea combined with the foliar spray of humic acid, water soluble fertilizers, and liquid micronutrients in rice. So these increased the increased uptake of rice crop for humic acid application was attributed to better use efficiency of applied nitrogen fertilizers in the presence of humic acid, and um, uh, and the highest nitrogen use efficiency also noted in that combined application of foliar spray, humic acid, and liquid micronutrient treatment. So, in conclusion, this study concluded that application of fertilizers based on the STCR IPNS NS urea and foliar spray of humic acid, water soluble fertilizer, liquid micronutrient at three different stages, and had increased the growth parameters and the yield of rice here at tune of 15.6% of rice grain yield than the conventional practice. So the treatment was found economically viable nutrient management options on farmers' field and Thamarabarani command area. Thank you, ma'am.
Okay, thank you, Ravina. Any questions from the student participants or the students who are attending offline? Any questions from the students who are attending offline? Okay. Then from my side, Navina, what do you mean by STCR IPNS? Can you just briefly narrate what is STCR IPNS?
It's a soil test crop response and integrated plant nutrient management system, ma'am. It is by analyzing the initial availability of the NPK values, we can calculate that how much of fertilizers were applied for the crop needed.
What was the target that fixed for this particular experiment?
Ma'am?
Yield target.
Yes, ma'am.
So applied SSR IPMS nitrogen as urea now. So what is
Yes,
the
ma'am.
yield target is available?
I can't hear, ma'am.
No. SSR takes into account yield target, isn't it? You used the equation, right? Huh? Have you used the equation?
Yes, ma'am.
In that there is a component T that indicates target. So how much target you have calculated the fertilizer dose and applied in this experiment?
Which?
Yield target, ma'am. See, okay, you are
Ma'am,
testing.
I can't get the questions clearly, ma'am.
Yeah, clearly, I'm just again telling what was the yield target you have fixed in this experiment. Because this SSCR works on yield targeting.
Yes, ma'am. 7 tons per hectare.
That's what this answer only I
Yes, ma'am.
got. That
Yes,
becomes
ma'am. 7
something
tons per hectare.
7 times per
The
hectare.
target
Okay.
yield of STCR IPNS is 7 tons per hectare, ma'am.
Okay, right. Okay, then
Yes,
thank you,
ma'am.
ma'am.
Thank you, ma'am.
The next one, Mr. Kadirvel, please. Kadirvel, are you online?
Yes, ma'am.
Kadirvel, you can proceed now.
Okay. Good evening to all. Myself, L. Kadirvel, research scholar in Kumbalur campus, Department of Soil and Water Conservation Engineering. Next slide. In most of the irrigation command areas, waterlogging and salinity problems very commonly affected by the field. So we reclaim that the waterlogging and salinity problem, subsurface drainage system will be installed in the field. That will be reclamation of waterlogging and salinity problem. My research is parallel drain subsurface drainage system. It's a method to lower the depth to groundwater table level below the root zone and create the favorable environment for the crop growth. Uh, this system involves in perforated PVC drain pipes will be installed in the below the ground surface. Um, that will be in perforated manner. That will be enrolled warped in the coir pit for around the PVC pipes and installed in the below the ground surface, and after that filling the soil and draining the water in the field. So alleviating the waterlogging problem in the uh, waterlogging condition area. So my research crop is selected in paddy. Varieties PPT 5204. The paddy field are in wa- uh, regularly in waterlogging condition. My research area is selected in Sembarai village. Uh, this is located in Tiruchirappalli district, Tamil Nadu, and my research areas carried out in laid out in 0.144 hectare, and lateral drain in between the two lateral drain pipes, the spacing will be provided. The spacing will be selected on based on steady state input equation. That equation based on lateral drain spacing will be provided, and drain diameter uh, selected based on the crop and cost of the purchase. And after that, lateral drain spacing selected in 7.5 meter spacing, 10 meter spacing, 12.5 meter spacing, and two drain depths we have selected 60 centimeter and 80 centimeter drain depth. The PVC perforator pipes at 63 mm will be selected for the warped around the coir pith envelope material, and lateral pipes will be providing the slope of 0.3%, and uh, lateral pipe will be connected to the inspection chamber, and all the inspection chambers uh, connected to the collector pipe. The collector pipe will be uh, slope on 0.6%. The research design is factorial randomized block design with three replication. The treatments is 7.5 meter drain spacing with a 60 centimeter drain depth drain, S1D1, 10 meter drain spacing with 60 centimeter drain depth, S2D1, 12.5 meter drain spacing with 60 centimeter depth of drain, and 7.5 meter drain spacing with 80 centimeter depth of drain, 10 meter drain spacing and 80 centimeter depth of drain, 12.5 meter drain spacing and 80 centimeter depth of drain, and also control plot. Next slide. And we have collected the soil samples in before the installation of the subsurface drainage system, and regularly before the installation collector samples, and 30 crop growing periods, 30 days after transplanting, 60 days after transplanting, 90 days after transplanting, 120 days after transplanting, and after harvest we have collected the soil sample and analyzing the some of the soil parameters, and what will be the result will be indicated uh, showed in the tables and graphs. First, soil parameters is soil pH we have analyzed, and all the treatments, each treatments we have selected soil samples, and it will be 7.5 meter drain spacing and the 80 centimeter drain depth will be uh, reducing level very lowerly. So that treatment is best level in the soil pH. It will be indicated in the graphs like this. Next slide. Next, soil EC we have analyzed, and it will be it is also reduced in the 7.5 meter spacing and 80 centimeter drain depth, because soil pH and soil EC, why it will be reducing level in because the drain water will be more in that treatment. So the waste forming materials and soluble materials, sodium and bicarbonate soluble in the drain water, because the soil pH and EC will be lower in that treatment. Next slide. Next slide. And also soil exchangeable calcium and soil exchangeable magnesium, sodium, soil exchangeable magnesium will be titrating procedure. That also collecting the soil samples, and it will be lowers in the 7.5 meter drain spacing and 80 centimeter drain depth. The value, very lower value is 17.86, and magnesium also lowering in the 7.5 meter drain spacing, and sodium and potassium also we have analyzed. That value is 12.87 in the 7.5 meter drain spacing and 80 centimeter drain depth. Like next slide. Based on the result, the parallel drain subsurface drainage system, 7.5-meter drain spacing and 80 centimeter drain depth has recorded in the considerable reduction in the soil characteristic values. Hence, it is recommended in that study area. So, generally, the cost of the installation of the parallel drain subsurface drainage system can be justified by increased paddy yield and total income. Therefore, it is concluded that adoption of subsurface drainage system increases the profitability of paddy field and improves sufficiency in food grain production. Thank you.
Thank you, Kadirve. Now the paper is open for discussion. Any questions from the student side? Kadirve, one question from my side. You said that that's 3.5 meter drain and 80 centimeter that has recorded higher yield and profitability in your last slide. Can you tell how much yield that you have recorded or the percentage increase in yield?
Ma'am, it will be recorded in 5 tons, ma'am. 5 tons nearer that recorded in that field.
No, you have completed the study, no?
Yes, ma'am.
The study is over, that only you are presenting. So
Yes.
it's a statement just to quantify and tell how much you got in the best treatment or what was the percentage increase in yield over your control.
Percentage.
And also you are telling that there is a good economy.
Yes, ma'am. Ma'am, it will be regularly farmers followed in without a subsurface drainage system. That will be indicated in uh, uh, above the yield level is 5% will be increased, ma'am. Regular followed procedures farmers mean my subsurface drainage system provided that means 5% will be increased.
Any more questions? If no more questions are there, I thank Mr. Kadivai for his presentation. And the next one is Nishala. Ms. Nishala, we can start with this.
Nishala madam is going to present in the offline one. And before that, we have to complete the online session, the OVM.
Okay. Nishala is going to present in person this week. So let me invite Ms. Ovia for presentation.
ஓரெல்லாம் இது மாதிரியே தெரியல பண்ணிட்டே
Hello, ma'am. Yes, ma'am. Am I audible, ma'am?
Yes, Ovia.
Yes, ma'am. Good evening to one and all present here. Myself, G. Ovia, doing first PhD in Department of Agriculture and Microbiology. I'm here to present my master's research work on exploration of rhizobial and non-rhizobial endophytes for salt stress alleviation in groundnut. Next slide, please. One of the important abiotic stress which our crop production facing worldwide is salinity. Salinity is increasing day by day globally due to various reasons. Some of the reasons are due to primary salinity and secondary salinity. Primary salinity is due to natural causes, and the secondary salination involves a human intervention like irrigation with salt enriched water, etc. Legume crops are sensitive to saline stress. No, the previous slide. Please, the previous slide.
Legume crops are sensitive to saline stress, in particular groundnut, which is known as the king of oil seed. It is also susceptible to saline stress, thereby its yield have been reduced due to salinity. There are various soil stress mitigation methods like breeding, genetic engineering method, chemical method, uh, and also use of biofertilizer. Use of bioinoculant for soil stress is an effective and eco-friendly approach. Thus, the aim of our study is to select the effective halotolerant plant growth promoting bioinoculant for alleviation of soil stress in groundnut. Next slide. Endophytes. Endophytes are microorganisms that colonize the healthy plant tissues, having a symbiotic relationship. Plants usually contribute various niches to endophytes. One of them is nodule. Our senior research work was about isolating the nodule endophytes. They have isolated up to 98 endophytes. In our research, we have selected 10 best endophytes, of which 4 are rhizobial and 6 are non-rhizobial endophytes. They have various plant growth promoting mechanisms like iron homeostasis, increasing the antioxidant activity, osmolyte accumulation, nutrient solubilization, etc. Next slide. So based on our study, we have designed the three objectives. First one is the screening of rhizobial and non-rhizobial endophytes for plant growth promoting under salt stress. Based on the result, we have selected four endophytes for their study on modulation efficiency, root exudate pattern, and their effect of salt stress alleviation in groundnut and under pot culture study. Next. Here comes the result subjective. It is a screening of salt tolerant level of endophytes. The salt concentration range was fixed from 0 to 15 percentage. Among all the 10 endophytes, they are capable to tolerate up to 5 percentage initial concentration, above which its growth started to decline. Next. So that 3 and 5% salt stress have been fixed and their plant growth promoting traits were analyzed under normal 3% and 5% salt stress condition. First graph shows the indole acetic acid production of all the endophytes under normal and salt stress condition, among which rhizobium fuscens are to have shown to produce indole acetic production, indole acetic acid production even under 3% salt stress condition. Next comes the ACC deaminase activity. The previous slide. Next graph shows the ACC-DMPase activity. Here, ethylene, due to abiotic stress, ethylene stress will be resulted. In order to overcome that, ACC-deaminase activity will prevent ethylene-induced stress. The organism which is capable of producing ACC-deaminase even under salt stress was found to be rhizobium fusion. Solstices have been reported to inhibit the uptake of essential nutrients by the plants. So that nutrient solubilization efficiency was analyzed for all the endophytes. The endophytes like the Bacteria dispersa, Rhizobium fuscens, Bacilloli, Bacillus speculensis have shown to solubilize nutrients under 5% salt stress condition. Next slide, please. Endogenous osmolyte content analysis. Microorganisms have developed various strategies to adapt fluctuating osmotic condition. One among them is the synthesis or accumulation of osmolytes. We have analyzed three osmolytes like proline, glycine, betaine, glucose. The organism like the Rhizobium fucens, Bacillus speculensis have shown increased osmolyte accumulation under 3 and 5% stress in proline and glycine betaine. Comparing proline and glycine betaine, glucose accumulation was found to be lower. Among the endophytes in glucose accumulation, uh, Bacillus speculensis and the Bacillus altigenesis have shown higher glucose accumulation under 3 and 5% salt stress condition. Next slide. We have analyzed the morphological characterization of endophytes under normal and stress condition. First and third column shows the organism under normal condition, and the second and fourth shows the organism under salt stress condition. We can see the difference like uh,
presence of wide spaces, accumulation of granules, and elongation of sizes between the normal and solstice condition. These wide space and the elongation of size have been reported as the adaptation mechanism by microbes to tolerate the solstice. Right. And based on the previous results, we have selected 4 endophytes, 2 are rhizobial and 2 are non-rhizobial endophytes, and we have decided the treatments like the single inoculum and the combined inoculum, and then the ninth treatment is a control. And we have studied its effect on the nodulation. Uh, Salinity have been reported to reduce the nodulation capacity by the in plants. So we have explained the nodulation way in Leonard jar experiment. The two pictures shows control and the plants treated with the rhizobium fuscens and the Bacillus speculensis. The nodule have been present in the endophyte treated plant, and it is completely absent in the control. And the results of combined inoculum have a number of nodules. Next slide. Have analyzed the root pattern by the endophyte treated plants in the saline soil in conducting in funnel flask experiment. This Venn diagram shows the root X-ray compounds comparing the control and the endophyte treated plants. In the endophyte treated plants, we have obtained the compounds having many plant growth promoting activities like a phenol, succinic acid, acetic acid, etc. Next slide. Next comes the pot culture study. The saline soil, which was collected from the Agrahara Samakulam, Annu, Coimbatore district, it was collected and sterilized and used for the pot culture study. 9 treatments were designed, which is completely randomized, and 3 replications were performed. The results the like biometric parameters like shoot length, root length, dry weight were noted down at 45 and 90 days after sowing. The results obtained show that the combined inoculation have shown the better biometric observations. In specific to that, the T surface, that is the rhizobium fusus and the Pantoia dispersa, have shown the significant effect on root length, shoot length, and dry weight. With regard to pod number and nodule number, the same treatment have shown the higher pod number and nodule number after 90 days after sowing. Next slide. This is the post-harvest biometric observation. The same results were observed that the combined inoculum have provided a significant impact on the root length, shoot length, and nodule number. Next, next slide. We have analyzed the antioxidant and and that's all. Only 2 slides. We have analyzed the antioxidant activity and osmolyte production in the leaf samples. The antioxidant activity, catalase and superoxide dismutase have found to be higher in the plants co-inoculated with the Pantoia dispersa and the Rhizobium fusion. With regard to osmolyte accumulation, proline, glycine, betaine, and ribulose accumulation was also found to be higher in plants treated with Rhizobium fusions and Pantoia dispersa. Next slide. Post-harvest macro and micronutrient content was analyzed in the saline soil, and the research shown that the endophyte treated plant soil have shown the increase in nutrient content compared to the not uninoculated soil sample. Next slide. So finally, we have come to the conclusion that two endophytes, Rhizobium pixans and Panthoiodispersus, have shown a significant impact on all the aspects of salt stress alleviation in groundnut. So, in future, it can after the field study, it can be developed as a bioinoculant, so that we can use it as biofertilizer for a salt stress alleviation in groundnut. Thank you.
The topic is open for discussion. Any questions?
Okay, just to tell the criteria for a saline soil. How will you characterize a saline soil? What are the parameters to describe
We
the
have
saline?
we have tested the physiochemical parameters, electrical conductivity and the pH, ma'am. The electrical conductivity was found to be low.
Values.
Yes, ma'am. pH is 8.4 and the EC is about 2.3, ma'am.
That is the saline soil. Or you are telling about the soil that you have chosen for the study. What is the ECT of the soil that you have chosen for the study?
Ma'am, EC is about 2.3, ma'am.
The soil.
Yes, ma'am. And the pH is 8.4.
Have you consulted any of the soil scientists before proceeding with the work?
Yes, ma'am, we have considered.
If you are consulted, most soil scientists would have told you the soil with 2.4 is a saline soil, right? And one more thing is, regarding the pot study you have shown a photo now, normally in any pot study, the number of plants that are maintained in the pot should be uniform, right? But it was not in your study, okay? And so the basic itself is there is a major flaw. Please correct it.
Yes, ma'am.
Have you analyzed for the EC of irrigation water?
No, ma'am.
Like without water, how the plant will grow, and any amount of salt contributed to irrigation water should also be taken into account.
Okay, ma'am.
Okay.
Thank you, ma'am. Ma'am? Yes, ma'am.
Sorry. You have used saline soil for what study or what? Normal or saline soil?
Ma'am, saline soil we have collected from the field, ma'am.
Okay. What is the role of the non-rhizobial endophyte? Rhizobial endophyte is fixing nitrogen. What is the role of non-rhizobial endophyte? Any idea?
Ma'am, nutrient solubilization and acetic acid, indolactic acid production, like both of them together contribute for plant growth promotion, ma'am.
Because one of your slides you have mentioned that 0% sodium chloride only, that is without salt only, they do the nutrient solubilization, phosphorus, zinc and silica. So when you increase the salt concentration and that way in saline soil, how do they, any idea?
Ma'am?
One of your slides, you have shown the nutrient solubilizing ability of different strains, in which they fix only in the absence of sodium chloride. They solubilize only in the absence of sodium chloride, not with 3% or 5%. There is no nutrient solubilization in the
Ma'am,
presence of sodium chloride. Yeah.
yes, ma'am. We have done the analysis, presence and absence, ma'am. We have selected only the organism with in the presence for salt stress, which solubilize the nutrient. We have selected for the further studies.
Okay, thank you. There are no more questions. Thank you, Gopi.
Yes, ma'am. Thank you.
Yes, Ms. Ishwara, you can proceed. Good afternoon all. I am Nishala, Department of Meteorology at TNAU, Coimbatore. Today I am here to present my presentation on prevalency and toxicity of different phytochemicals against cigarette beetle. Cigarette beetle is one of the most serious pests actually. It has a wide pest range it has a wide pest range and the main control depends upon the phosphine. But phosphine directly applying phosphine on the storage waste is have many undesirable effects on the human health. So development of eco-friendly tactics are very much important for controlling the storage waste. Nowadays, essential oils and their components have gaining much more importance because as they can manipulate the insect behavior and have insecticidal properties. I have used the cigarette beetle culture was maintained in turmeric and coriander in laboratory conditions. These are the treatments used for the study. These are procured from the sigma outreach and used for the bioassays. Next. Prevalence study was done by area prevalence method given by U and by U. Here filter papers were cut into diameter of paper plate, and each is cut into two halves. And first half different doses of treatments were applied, and next half acetone is applied. This will act as control. In each application, 20 insects were released at the center of the petri dish, and percentage repellency was calculated by this formula. And here NC is insect present in first control half, and NT is insect present in the treated half. Next. Almost all these compounds showed repellency, but cinnamaldehyde and eucalyptol next slide. Cinnamaldehyde and eucalyptol showed a great degree of repellency, even at lower concentrations, followed by glycine. But in case of citral and beta-citronellal, at very low concentrations they have acted as attractants, but by increasing concentration they have acted as repellents. Next. Toxicity assays were studied by using topical application, surface treatment, and fumigant assays. In topical application, different doses were applied on the eggs. In each application, 20 eggs were taken, and observations were taken after 10 days of treatment. Next. And the mortality rates were subjected to profit analysis to get LD50 values. In this, transinamide exhibited a highest toxicity with low LD50 values, followed by eucalyptol, and least toxicity was observed by geraniol. Next. Same pattern was followed in case of drugs also. Next. In case of adults, next. In case of adults, highest toxicity was observed when treated with transinamide and followed by eucalyptol. And citral exhibited lowest toxicity. Next. Surface treatment, penetrated surface treated with different doses, 5, 10, 15, and 20 and 25 microliters per centimeter square, and then shade dried. And eggs, grubs, adults were released in the petri plate, and their mortality was checked. Next. There was no effect on eggs, but in case of grubs, cinnamaldehyde showed the highest toxicity, followed by eucalyptol, and least by citral. Next.
In case of adults, next. In case of adults, trans-cinnamomatoid and eucalyptal were exhibited highest toxicity, and the least was exhibited by beta-medicine. Next. In case of fumigant toxicities, the assets were done in 500 ml bottles, and the treatments were imposed at the bottle cap. 500 microliters of different treatments of 1%, 2, 3, 4, and 5% concentrations were used, and mortality rates were taken. There was no effect on eggs. Next. In case of drugs, trans-cinnamomatoid exhibited highest toxicity, followed by beta-medicine, and the least was exhibited by Citral. In case of next. Next. In case of adults, trans-cinnamomatoid exhibited the highest toxicity and followed by beta-medicine, and least was exhibited by geraniol. In case of contact toxicity, trans-cinnamomatoid is followed by eucalyptal, but in case of fumigant toxicity, beta-medicine was replaced by eucalyptal. Beta-medicine have some highest fumigant, more fumigant toxicity than eucalyptal. Next. This was a new methodology. In fumigant toxicity were studied at different heights. At two heights, they were evaluated at 5 centimeters and 15 centimeters. In these two experiments, in first experiment, 200 microliters of 50,000 ppm were applied at the bottom. In the second experiment, the same concentration were applied at the top. But the results showed in the first experiment, cinnamomatoid showed 56% mortality at 24 hours after treatment. But at 50 centimeters, there very less mortality was seen. And not only for cinnamomatoid, almost all the compounds have no significance at 50 centimeters height. Next. When treatments were imposed at the top, cinnamomatoid, all the compounds showed mortality at both the heights. Cinnamomatoid is the highest and least followed by beta-medicine, and least mortality was exhibited by thymol. This difference in mortality may be due to the molecular weight of these compounds. If we compare these molecular weights of these compounds to aluminum phosphide, this aluminum phosphide has very less, that is 34 molar mass, but all these compounds have very high, that is more than 130. So these compounds have tendency to move from top to bottom, but they have no tendency to move up when applied at the bottom. Next. Finally, I will conclude that trans-cinnamomatoid has much toxicity and highest repellency activity, but it has some stability issues. As this is a phenyl propanoid, this will immediately convert immediately oxidizes into cinnamic acid. But cinnamic acid is also having toxic effects. But according to the literature, trans-cinnamomatoid is slightly more toxic than cinnamic acid. To make it more stable, we can add antioxidants, or we can go for nano-encapsulation. My future line of work is on nano-encapsulation of trans-cinnamomatoid. Thank you. The questions are invited on this paper.
You mentioned that in trans-cinnamomatoid there is much toxicity and more potency. And also you mentioned about aluminum phosphide. Aluminum phosphide is a check for your study?
Actually, I have studied at 2 meters height. For 2 meters height also it reached very fast.
and 100% mobility for aluminum phosphide.
Can you explain why? Can you explain why?
Actually, the structure of the aluminum phosphide has no double bonds and it easily evaporates into phosphine gas. This phosphine gas is, have very much higher loss to the insects. Very much promised immune.
What is the mechanism of action of the eucalyptus?
Eucalyptus is actually having good contact oxygen, but in case of chemical toxicity, beta medicine is replacing eucalyptus because maybe that molecular weight and the structure dispersion is all maybe maybe the reason.
You are not you are not characterized reason for no study.
Ma'am, next stability and further I am going to study.
Okay, good evening, one and all. Dear coaches, Dr. Shanti, Joseph Swanson, Dr. Chemistry, Dr. Kalachiri, Professor Practice Microbiology. And there were two lead papers in this session. And with guest speakers, one from Vivo and another from Vivo, and followed by 5 presenters. And all the 5 presentations were good. And each one paper has been focused in a different angle. So we thank all the presenters for their presentation and participation in this mode, and also all the offline and online participants who have participated and raised the occasion. So with this, the oral presentation comes to an end, and now I hand over to Dr. Kumar, Professor of Agricultural Microbiology, and Dr. Sara Bano, Professor of Environmental Sciences, and Dr. Selvi, Professor of Science and Agricultural Chemistry, to handle the session for the poster presentation. Thank you.
Thank you so much, ma'am, for making this session successful for us. Thank you so much. So we come to the end of the oral presentation. Next, we will move on to the poster presentation. We request ma'am first to please take over. Very good evening, sir. So now we will move on to the poster session. Handing over the mic to you.
Okay. எல்லாரும் வந்துட்டாங்களா ஃபர்ஸ்ட் போஸ்டர்
Very good evening to all of you. Great pleasure for us to view the posters of the topic natural resources for enhancing food production. So there are about 44 posters which needs to be completed within 60 minutes. So that was the time given to us. If there are any clarification to be obtained from the poster presenter, we will call you. So otherwise, we will run through the poster and then decide based on the content available in the posters. So we were asked to check the clarity, flow of presentation, novelty of the contribution to the field, and the ability to interact. Of course, we may not be able to interact with you, to all the participants, but we will try to speak to you based on the doubts or clarification that arises from the poster. So with this, maybe we can go one by one. So if anything is to be clarified.
We wish you all the very best for your posters. Please stick on to the timing so that you get equal time distribution to all the participants. Thank you.
Yes, ma'am. Full zooming, ma'am. Lights off on.
Dear students, so just there is a change in the way that we are going to evaluate. I think all of the poster presenters can have 1 minute, and you just give the title and maybe the novelty of your work just within a few seconds, and then tell us the inference. So whatever you want to highlight in the poster, so you just make it a point to concise it in 1 minute, and then you can present to us based on that we will evaluate. Thanks. I think the first poster is by Devi. So Devi, are you there?
Yes, ma'am. I'm here.
Okay, so we will give maybe 1.5 minutes for you because we just have to prepare and speak to us. So you get just an extra time, but for the others, yes, you please speak on to 1 minute, please. Thank you.
Ma'am?
We can start.
Posters, I want to share the posters, ma'am.
It's already shared. This week.
I couldn't see the poster, ma'am.
1 second. Okay, you just see your poster.
Okay, sir. I'll start the presentation, sir.
கொடுத்துட்டீங்களா ஷேர் ஸ்க்ரீன் Is it visible now?
Yes, ma'am, it is visible. Visible, ma'am. Shall I start the presentation, ma'am?
Yes, you can start.
Okay. Good evening to one and all present here. I'm K. Ragadevi, doing third
PhD in Department of Crop Physiology. Now I'm going to present about my research topic on altering the physiological response of compact cotton.
Can be a little bit louder.
Okay,
And
ma'am.
just you can just explain your research and conclusions. That is enough.
Okay,
Because you have
okay,
to
ma'am.
speak on 1 minute only.
Okay, ma'am. As we all know, cotton is the most important cash crop and it plays a major role in improving the agricultural economy of the country. But recently, according to USDA FAS, there is a 2.4% reduction.
Hello,
Ma'am, yes, ma'am.
We are not able to hear your voice properly.
You're just talking slow, you just be a bit louder or just put your speaker.
Okay, ma'am. Okay, ma'am. As we all know, cotton is the most important cash crop and it plays a major role in improving the agricultural economy of the country. But due to some climatic factors and unfavorable environmental condition, there is a significant drop in cotton productivity. There is about 2.4% reduction according to USDA FAS. So, and this fluctuation, it needs a new innovative research to increase the cotton production and also to stabilize the cotton productivity around the world. So here comes the role of biostimulants. These biostimulants are the potential resources and they are the trend for the destiny to improve the production. And they plays a major role right from breaking dormancy till the harvest. And the effects of these biostimulants, they add up to the improved and balanced growth and development of the crop. So with this background, this present experiment was taken to study the impact of biostimulants on growth, physiology, and yield in the compact cotton. And the field experiment was executed in randomized block design with 6 replicates in the newly released compact cotton variety Core 17 with 7 treatments. The treatment details include Enterobacter hormachi at 2 different concentration. One is 10 kg per hectare and 20 kg per hectare. And next one is Glomus intraradices. This also at 2 different concentration at 10 kg per hectare and 20 kg per hectare. And Bacillus subtilis at 10 kg per hectare. And consortium of VAM with vitamins and amino acids at 10 kg per hectare. And these inoculants were treated after 6 days after thinning to the cotton seedlings, and the observations were recorded. So coming to the results, the leaf area and photosynthetic rate in the treatment consortium of VAM with vitamins and amino acids, they recorded the highest leaf area and the photosynthetic rate. And due to the presence of more leaf area, there will be an accumulation of more photosynthetic pigment. Sir,
Thank you. Thank you.
thank you.
Next, 41. 41, please. Your time starts now.
Control L. Okay.
41. Are you there? Venkatesh? Venkatesh is not there. Gopal?
He didn't give the poster. No?
Venkatesh is not, he didn't give the poster.
He didn't upload his poster.
Venkatesh.
Parthiban.
Venkatesh?
So Parthiban is there. Parthiban, he uploaded the
Incorrect.
What happened?
They didn't connect it.
They didn't connect. But their poster is available.
Poster is
Venkatesh.
there, unless it's not available.
No problem. Venkatesh poster is there?
No, no.
No, not there.
Are you there? Focus on it, sorry. Hello?
Good evening, sir.
Gokul, are you online?
Yes, ma'am.
Okay, then you start presenting.
Okay, ma'am.
1 minute.
Okay, ma'am. Good evening, everyone present here. I am going to present
Yeah,
poster on
Please be louder.
Okay, ma'am. I am going to present poster on assessment of rhizosphere microbiome in reed plants using metagenomics. We have collected 5 reed plants along the Noyal River Basin, which were identified as Colocasia esculenta, Xanthosoma, Cana, and Saccharum spontaneum and Typhodomengensis, and they were used to treat the wastewater from the paper mill. And the best treated plants such as Colocasia esculenta, Cana indica, and Typhodomengensis reduced BOD and COD drastically with significant difference. So these plants were selected to extract the rhizosphere soil to assess the microbial diversity in the rhizosphere. And the results of the metagenomics shows that maximum percentage of bacteria were present in the these 3 reed plants, and the alpha diversity also estimated. The observed species of from the Cana indica, Colocasia esculenta, Typhodomengensis were written. And
ever since the beginning. Ever since the beginning of agroecosystems in the Nilgiris, soil carbon pools has decreased from 75% in the past 100 years. And hence, a detailed soil survey was carried out in the entire Nilgiri Biosphere Reserve to know all the physical, chemical, and biological properties to figure out the soil carbon stock. It has been found that soil organic carbon stock decreases along with the depth in all the regions. So this opens the way for carbon farming. Since the soil is having a reaching limit of about 150 tons per hectare of carbon, so by doing the carbon farming, this will enhance the carbon in the soil. So the carbon which is acting as a source of the heat in the atmosphere can be brought down to increase the organic carbon. Thereby, it will provide the energy to the soil microbes and hence enhance the soil biodiversity. So together with the organic carbon and biodiversity, a better physics can be maintained, and thus the carbon farming can be acting as a sink to the global carbon, and hence it will combat the climate change. So with the present research, I can give 2 conclusions. Since India is being a signatory to the Sustainable Development Goals, this present research can mitigate the Sustainable Development Goal number 13, which is the climate action. And second is, since the farmers is giving more options for the carbon credit, so this will help in enhancing doubling the farmers' income, which is a government ambitious project. So doubling the farmers' income and the carbon is my conclusions. So keep soil alive and protect soil biodiversity, which is the pledge of World Soil Day in 2020. And Potash has most slope and altitude is regional. In case of Potash region, there is no amount of forest, and the farmers is going to the organic farming when compared to the Aparna.
Good evening, sir.
Yes, please.
Good evening to all present here. I am going to present my part of my research work.
You all have to speak louder. Come on, you just speak a bit loud.
Okay. My topic is about the comparative study of epiphytic and endophytic bacteria associated with the seeds of Zea maize. I specifically chosen the crop maize because it is third most important crop after rice and wheat, and I have chosen the seeds of microbial work on the seeds of Zea maize because seed harbors some diverse group of bacterial communities that are present both present on their surface and also reside inside the tissues. Seed tissues. The microbial communities which are present on the surface of the seeds are called epiphytic bacterial communities, and the microorganisms which are present inside the tissues, these seed tissues are called the endophytic microorganisms. I have compared both the population of epiphytic and endophytic bacteria of the maize seeds and how it get influence the maize health, plant health and its function. And in my topic, I have chosen the 3 different maize seed types. Why? Because the seeds may act as a vector for the microorganisms which carried across the generation. For that purpose, I have chosen the one hybrid ruling variety of maize that is maize CoH6, hybrid CoH6. Hello.
Thank you. GP?
Yes, not uploaded.
Okay, that's Jayaprakash. Sorry.
Hi, sir. Hi, sir. I'm there.
Yes. You have 1 minute time.
Okay, sir. Good evening to all. Now I'm going to present my poster on topic crustacean meal as a fish meal replacement in aquafeed for improved yields in aquaculture. And my primary objective is to convert the crustacean wastes, particularly the shrimp waste and crab waste, which contributes around 60% of the total landings in harbor. So we can utilize these wastes into a feed for fish to improve the yields in aquaculture sector. And my research work was carried for 60 days, in which 3 diets were prepared and fed with the fishes. And at the end of the experiment, the result shows that shrimp waste and crab waste feeds performed better than the normal control feed. So we can utilize this shrimp waste meal and crab waste meal as a feed for fishes, which will give better performance than the normal control diet. Thank you, sir.
Thank you.
Thank you, sir.
Thank you. Next, Kalai Selvi.
Hello, sir. Can you hear me?
Yes. Yes, we are.
Being a student of soil science, let me kickstart my talk with the saying there can be no life without soil and no soil without life. They have evolved together. I'm Kalai Selvi K, pursuing my master's research at TNAU Coimbatore. Indian Council of Agricultural Research, in collaboration with State Agriculture University, initiated All India Coordinated Research Project on Long-Term Fertilization Experiment LTFE 1972 in major agroclimatic zones of the country, including TNAU, under which the intensive continuous cropping systems, a detailed investigation was contemplated to evaluate the long-term effects of fertilization on rhizosphere profiling with reference to P dynamics has been carried out. The objective of my study was to record the effect of inorganic and integrated nutrient management on the changes in the available phosphorus status in the rhizosphere and non-rhizosphere soil during the cropping period in maize. With regard to the experimental details, the field experiment was conducted in
Hello, sir.
field number 36
Sir, that
of the Eastern Block in
presentation
TNAU
over, sir,
Coimbatore.
for me. Sir, they give 1 minute only, sir, time to present.
In PK 100. Hello? Hello? Ma'am, was I audible? Ma'am, should I repeat or?
What's your final conclusion, ma'am? What's your final conclusion?
Ma'am, the enhanced, yes, ma'am, my final conclusion is that the enhanced levels of available B was due to the conjoint use of manure and fertilizer promoted the recycling of nutrient and transformation added, ma'am, which ultimately led to different labile pools in the continuous intensive cropping system, ma'am.
Okay.
Thank you.
Sandeep Kumar, are you available? Yeah, Sandeep Kumar.
He's not on, no?
Good evening everyone present here. My topic is effect of integrated plant system on yield response, uptake and the chemical level of soil available NPK on intensive soil. First of all, my introduction is the panel is one of the small millet is the important crop among the millets. It is also the fourth important food production Asia. Further, his methodology is I implemented the SPGS crop response methodology and scale was split into 3 splits and also fertilizer treatment also applied in the 6 different aspects. Further, in this methodology, we have noted that strip 1 and strip 2, strip 3, but then we applied a different combination of treatments. Further, we conclude that yield was increased was strip 1 to strip 2 compared to value is increasing, and also limited response also the value increased from strip 1 to strip 3 was high. And also here is the nutrient response applied better nutrient response also is was high.
Thank you. Venkatesh.
Hello, Venkatesh. You can start presenting. Hello. Are you online? Venkatesh, are you online? You can online.
லாபியில் பார்க்கட்டுமா? லாபியில் பார்க்கட்டுமா? வெங்கடேஷ் சாரை லாபியில் பார்க்கட்டும்டாங்க.
இருக்காங்களா?
It's not there.
Okay.
Hello, madam. Good evening, madam.
Yeah, you just be a bit louder when you present your poster.
Okay, ma'am. I'm going to present about effect of Nurata potash and Salvita potash on nutrient uptake and yield of green gram. The pot culture was taken with different levels of MOP and SOP. The same treatment were followed in field experiment. Coming to the results, a highest uptake of NP, Ks and yield were recorded in 40 kgs of sulfate of potash in both pot and field experiment. T10 is statistically on par with the T6, that is 40 kg of MOP. Cost of SOP is higher than the compared to the MOP. I concluded that application of MOP 40 kg per hectare recommended for the maximum returns to the farmers. Thank you, ma'am. Hello.
John. Thank you, Mark. Thank you.
Thank you.
John.
Yes,
John.
sir.
Yes.
Yes, sir. Hello.
Please proceed.
Okay, sir. Ma'am, I'm waiting for my poster.
You can be a little bit louder.
Okay, ma'am. Okay. Evening greetings to everyone. TNAU tomato hybrid CO3 is one of the most widely cultivated tomato cultivar in the state. My master's research was to find out the effect of water-soluble fertilizers on the nutrient uptake pattern in this tomato hybrid, which was planted in pots at Department of Soil Science and Agricultural Chemistry. And with respect to the vegetative parameters like the plant height and number of leaves, it was found that fertigation with NPK at the ratio of 2:1:1 gave the maximum results. This may be due to the positive effect of increased application of nitrogen, and this study can be made useful to optimize the fertilizer use so that the excess and under application of fertilizers can be avoided. Thank you. Yes, ma'am. Ma'am, I can't hear you.
Hello, are you online?
Hello, ma'am. Hello. Ma'am, I'm Ajila, ma'am.
Hi.
Ma'am, shall I start, ma'am?
Yeah, you can proceed.
Okay. Good evening, everyone. My research was conducted on soil test crop response integrated plant nutrition system on drip fertigated tomato. We conducted the field experiment in Kuppanur village of Coimbatore with 15 treatments replicated thrice. The pH and titratable acidity among the different treatments were compared, and the results indicated that the treatment STCR IPNS at 12.5 tons per hectare FIM at a yield target of 90 tons per hectare recorded the highest titratable acidity due to increased organic manure application and potassium fertilization. The increase in titratable acidity was parallel with decrease in pH. Since this treatment has increased titratable acidity, the pH was at its least, whereas the absolute control recorded the highest pH and lowest titratable acidity. Thus, I conclude that the pH and titratable acidity has negative correlation with each other. Thank you.
What is the normal range of titratable acidity in tomato?
It is from 3.5 to 4.5, ma'am. % citric acid.
Okay.
Thank you.
In the different treatments, can you have this line graph?
Ma'am?
I'm asking a question about your line graph. X-axis எல்லாமே treatments கொடுத்தீங்க எல்லாமே variables தானே எல்லாமே different treatments கொடுத்தீங்க. Can you have a line graph or which graph is suitable for this? A line graph or a bar graph? I know it's out of your technical section. Which graph is more suitable for your data?
Ma'am, I can't understand, ma'am.
You just go and check with the distributable. Thank you.
So next is Dharmendra, are you online? Parvati Sumari, are you online?
Yes, ma'am.
Can you proceed?
Okay, ma'am. Shall I start, ma'am?
You can start.
Good evening, one and all present here. I am Parvati Sumari, pursuing second MSc in the Department of Soil Science and Agriculture Chemistry. Here I am going to discuss about the enhancement of growth and physiological attribute by adapting STCR IPNS approach along with drip fertigation for aggregatum onion. As onion is a shallow-rooted and highly nutrient-required crop, balanced fertilization is an important requisite for achieving the high yield. The STCR IPNS along with drip fertigation will be the promising strategy to catch up with rising demand for attaining the desirable crop growth. With this regard, the field experiment was conducted in Kuppanur village of Thondamuttur with 15 treatments replicated thrice. Here I explained about the effect of this approach on plant height and SPAD value. It was high in the treatment of STCR NPK plus FIM at the rate of 12.5 tons per hectare, followed by STCR IPNS using biocompost. This was due to the slow and effective release of nutrients from both FIM and biocompost. As per this, and drip fertigation also satisfies the stage-wise requirement of the crop. So the desirable crop growth was attained by adapting this approach. So the SCCR IPNS plus drip fertigation will be the pressing priority for accomplishing the desirable crop growth and also by achieving the balanced fertilization. Thank you.
Okay, can you relate the treatment with reference to yield data?
Ma'am?
Can you relate your physiological attribute with your yield data? Space figure reading.
Yes, ma'am.
Can you relate the reading with the yield data?
I can't hear you, ma'am.
Hello. Now can you hear me?
Yes, ma'am.
You please correlate the physiological attribute that you have mentioned, SCMR, no?
Okay.
With that of yield data. Yield, onion yield.
Yes, ma'am.
Okay, thank you.
Ma'am, Didn't mention here, ma'am, about the yield.
Yes, ma'am.
You're online, Shahima?
Yes, sir.
You can start.
Good evening. Myself Shahimat Sohra. I am going to present the poster on the topic estimation of surface runoff from subcatchments of Karamade using SES cow number method and GIS techniques. Here, as we all know, surface runoff will cause depletion of soil and water resources, and it will in turn adequately affects the agricultural production. So here we can calculate the runoff both conventionally as well as using hydrological model. In this study, I have calculated the storm-wise runoff for 20 years duration starting from 2000 to 2019 using SES cow number model with the integration of GIS technology as well as remote sensing. And here for the three catchments of Karamade block, I have done the calculation. Three catchments, especially for Paraipallam, Mandraipallam, and Periyapallam. And here detailed methodology is given here in the flow chart, and the data I have used were rainfall data, soil data, as well as the land use which were prepared from the Google Earth image as well as from list 3 satellite data. And here in this table, the result was given that is corresponding to runoff for each year, how much rainfall for each year, how much runoff has produced for each catchment, like three mentioned catchments. And the major findings which I have obtained in this study are the maximum runoff it was obtained in the year 2011, and maximum runoff it has happened in the Mandraipallam catchment. And the major use of this study is we can prioritize the catchment while before we are planning for rainwater harvesting structure. Thank you.
Fatima, what to say? With the runoff, is there any specific methodology or what is the methodology you follow?
I have used a SES number which uses the land use, soil, and antecedent soil moisture content in the soil. So here if the soil is dry, means soil can hold maximum water, thereby we can reduce the surface runoff. So based on these all three factors like rainfall, soil,
Okay,
and
okay,
land use,
done.
we are
Okay, thank you.
okay. Thank you.
So, Sumaya Sultana, are you online? Sumaya,
Hello.
is it audible for you?
Yes, I'm here, ma'am.
Please share your presentation. Can you please start listening?
Shall I start, ma'am?
Please start.
Yeah, the study was conducted to evaluate some danger genotypes at 10 days interval.
Or you just be close.
Sorry, ma'am, I can't hear.
No, you can start listening. Let me know.
Hello.
Is it audible?
Yes, ma'am, it's audible.
You can present it, but please loud.
Four genotypes you have screen. It's Vellore local, Sivagangai local, Pantensha and Vilupuram local. From the results, it is seen that Sivagangai local recorded good germination percentage, good plant height, base diameter, whereas Vellore local recorded more biomass at 60 days after sowing. So in future crossing can be effected between the Sivagangai local and Vellore local for getting better recombinants from both parents to produce high yielding Danger varieties.
Balaji. Next is P. Gayatri.
Hello.
Next is P. Gayatri. Are you online?
Yes, ma'am.
Okay, you can proceed.
Good evening to one and all present here, ma'am. I'm going to present my topic is generation of soil fertility map of Guntulur village of Perambalur district. And first I have to collected the cadastral map of Guntulur village on survey and land records. They are giving the map for 2 or 3 maps. We want to stitch using with the method of map stitching in Voice Image Pro software. After that, the map was georeferencing and digitizing using with the software of AGSC software. Then the soil data or geocoding the nutrient database with the survey points. And after finally we have to generate the cadastral level soil macronutrient maps. They are very useful for knowing about the efficient basic information about the soil fertility status of an individual survey number, and also useful for monitoring the fertilizer schedule, which remains to be a sound specific technique for achieving the higher crop productivity. Thank you, ma'am.
Next is R.C. Prashant.
Yes, ma'am.
Okay, you can proceed.
Good evening to one and all gathered here. I'm Prashant, second PhD scholar from Department of Agronomy. This poster, I'm going to tell about the effect of crop geometry and nitrogen levels on multi-cut fodder sorghum. Here, as we can note, there's a growing demand for the difference between the demand and supply is developing gradually in the coming years. So we have to increase the fodder productivity in the existing land to sustain our milk production and animal productions. So here undertaken a study on intercropping of fodder cowpea in between the rows of fodder sorghum by adjusting the normal spacing to pad row spacing, thereby giving an additive series of intercropping. Here, the main plot consists of crop geometry and the subplot consists of the nitrogen levels, 3 nitrogen levels, 125%, 100% and 75% of RDF. As we can see, I have taken 3 cuttings for the experiment. In the first 2 cuttings, the sold fodder sorghum under 45 into 15 centimeters showed the higher growth parameters and the yield and the quality parameters. But in the third cutting, due to the beneficial effects of the intercropped cowpea, fodder cowpea, the yield and the growth parameters was positive for the S4 treatment. That is the pad row system of fodder and fodder cowpea. Okay, sir.
Next is Ashana. Yes.
I'm glad to present the topic titled Impact of Targeted Yield Model-Based Fertilizer Prescription on Physiological Attributes of Millet on Insecticide. First of all, my crop has raised the Eastern block, which is Perinacan Pallium soil series. taxonomy termed as verticostropher, and it is a mixed black calcareous soil. The experimental design carried out was randomized block design and consists of 10 treatments and replicated thrice. The treatments were STCR NPK alone treatments and STCR IPNS treatments with 3 different levels of yield target. The yield target were fixed based on the yielding potential of the variety. And the next treatments were blanket alone and blanket plus FIM, which is applied at 12.5 tons per hectare. All the treatments were compared with our formal practice and absolute control. In this, the Millet has 4 critical crop growth stages. That is vegetative, panicle initiation, grind filling, and harvest stages. The physiological attributes I presented here was spat chlorophyll meter reading and crop growth rate. Among all the treatments, the SCCR IPNS treatment with the highest level of yield target recorded the superior values. It was also parallel with STCR and NPK alone treatment with the highest level of yield target. The superiority of these treatments is mainly due to the balanced supply of nutrients. That is, the plants receive nutrients from both the inorganic fertilizers. Thank you.
Next is Bhavna. Bhavna, are you connected?
Yes, ma'am. Good evening to one and all present here. Myself, Jangam Bhavana, second PhD Agronomy. Nipping on crop geometry plays an important role in enhancing the yield of crop. Hence, an experiment was done in factorial run RBD with 2 factors. First factor includes crop geometries and second factor includes nipping both by manually and also chemicals by using chemicals like mepiquat chloride and chloromequat chloride. So coming to the results, the higher dry matter and the yield were observed with the line swing of 30 by 10 centimeters. And in case of nipping, it is with the spraying of mepiquat chloride to 50 ppm. This chemical inhibits the apical dominance so that more number of branches will be produced. This leads to the improved production in the yield of horse gram. Hence, it can be concluded that treatment combination of 30 by 10 centimeters along with the spray of mepiquat chloride to 50 ppm resulted in higher productivity in rain-fed horse gram. Thank you, ma'am.
I have got a small, just a suggestion to you and this person, Prashant. So I think when you present a poster, the very first criteria is the font size. Alright? So I think you just see your heading and also the font size of the treatment. It's not even visible in this big screen. So maybe next time when you present the poster, make sure that it is visible. So there is some criteria when you see there is a lot of space in the poster. So you have made the graphs and also the other things a bit larger. Okay, this is just
Next time I'll do. Okay, thank you.
Next is Mahesh. He will be presenting offline.
My first topic is based on influence of soil-based crop response based inorganic and organic fertilizers on quality of food. So there's a crop like use of fertilizer testing the soil is like taking medicine without consulting a doctor. So whenever we are going for SPGS based organic and inorganic fertilizers, the quality of fertilizer has been improved, like mucilage, protein and starch content. Not only the quality, but also the yield has been improved. This is mainly because when we are using organic and inorganic, there will be production of more organic acids that will tend to solidify your native nutrients, thereby increasing the quality, absorption of nutrients and quality as well as the yield. So whenever we are going for the balanced fertilizer recommendation, that is by SPGS and IPNS, the soil will be healthy. When the soil is healthy, the plant will be healthy. So altogether, we can go for sustainable agriculture. So this advantage of this SPGS IPNS is this inorganic nutrient will take care of the immediate requirement of the crop, as well as your organic fertilizer will take care of your future requirement of the crop. Thank you.
Next is Ashmita.
Good evening to one and all gathered here. My topic is on interaction effect of vesicular or vesicular mycorrhiza with different amendments on increasing phosphorus uptake. As we all know that the phosphorus is the most limiting nutrient. As well as its availability is very difficult for the plants, we have conducted the study to find out the best phosphorus management strategy in alkaline soil. We have conducted a pot experiment with maize with the following treatments, in which we have applied the phosphorus with the ammonia manure and air ammonia manure and UV test. All these treatments were tried with and without VAM to find out the synergistic effect of VAM with the different treatments, and the results conclude that the application of the phosphorus along with FIM in the presence of VAM increase the phosphorus availability as well as the mobility. This is because the FIM acts as a phosphorus solubilizer and VAM acts as a phosphorus mobilizer. Instead of applying a solubilizer or mobilizer alone, applying this both in a combined form can increase the phosphorus availability in soil as well as the uptake movement. This has been shown in the results in the form of table as well as in the graph. And this is what I would like to conclude from this poster. Thank you.
Next is Vinitha. Vinitha,
You can accept everyone, okay? Last. You have any technical to type message?
So, lobby level is from the
It's not okay.
next is Naveen Kumar. Next is Monisha Prakash. Are you online?
Next is Sen.
Sengathi Kumari, are you online? Next is Ranjit, are you online? Is it audible for you? Hello Ranjit, can you please unmute yourself?
minimize பண்ணுங்க அவர் அன்மியூட் பண்ணிருக்காரு மியூட் பண்ணிருக்காரு மியூட் பண்ணியிருக்காரு.
Yes, ma'am.
Okay, you can start presenting.
Hello.
You can start your presentation.
Hello, ma'am. Am I audible?
Yeah, but you can be a little bit louder. You're not audible. Yeah.
Okay.
Now you can start.
Good evening, one and all present. I'm going to present on the topic pollinator diversity in Moringa olerifera system in the red sand dunes of Thuthukudi district of Tamil Nadu. The red sand dunes is colloquially called Theric, which means the red desert, ecosystem present. Here we have placed Apis mellifera colonies in the Moringa field and recorded the pollination efficiency of Apis mellifera and recorded many diversity of majority of the pollinators in this ecosystem. Among the ecosystem, Apis serrana and Apis mellifera are the most predominant insects which actively participate in the pollination of Moringa flowers. Regarding the pollination efficiency index, the Xylocopa carpenter bees actively participate in pollination, but their abundance is less. After calculating the pollination efficiency, the pollinator rank was one given to the Xylocopa, and the next efficient pollinator was the Apis mellifera.
Next
hello.
question. Are you online?
Yes,
Yes, ma'am.
Conclusion. Apis.
Thank you so much. Your time is over. Okay, we are going on to the next presentation. So, Vishnu, you can continue your presentation.
Thank you, ma'am. Good evening to the gathering. I'm going to present about production and the characterization of polyhydroxybutyrate from Bacillus aemonsis. Polyhydroxybutyrate is a secondary metabolite which is produced under a starvation condition, which has the similar properties with the plastic which is available in nature. Using the methodology, I have collected soil samples from different locations and isolated the PHP producers by initial screening of Sudan black staining and Nile blue staining. Under the optimized condition using RSM methodology, I have isolated and purified polyhydroxybutyrate and confirmed using FTR and XRD method. Using FTAR, the corresponding groups which is OH and C double bond O and corresponding to the wave number of 3266, which is similar to the PHP which is available in nature. This PHB shares similar property with the petroleum plastic, so we can alternatively use this PHB as a bioplastic material for further use.
Vishnu, thank you. Your time is over. Because she has not uploaded the question.
It's not clear.
Next is Advantage. Are you online?
Thank you, ma'am.
Okay, you can present. Is it audible? Hello? Hello, Anika? Hello? Are you online? So next is Prakash and Bose. Are you online? Yes. Connected. Hello, Prakash. Hello, sir. Hello, sir. Is it audible? Next post.
So rise here, rise here,
Yes, sir.
Hello, Raisa.
Yes, ma'am.
Okay, you can start.
Ma'am, I'm present, ma'am.
Sure, you can start.
Hello, ma'am.
Is it audible?
Ma'am, no, ma'am. It's not audible. Hello, ma'am.
Raisa, is it audible?
Hello, ma'am.
Raisa, is it audible? Audible for you?
Yes, ma'am. It's audible, ma'am.
You can start your presentation.
Yes, ma'am. Good evening, everyone. In this study, 50 rice landraces were used to analyze the genetic divergence for grain size and weight, because grain size and weight are one of the major yield determining components. Mahan-Globus D-square statistic were used to group the genotypes into 4 clusters based on their divergence level. The genotypes in cluster 1, like Ilpaipu Samba and Garudan Samba, were recorded highest mean value for seed breadth, 100 grain weight, D-husk 100 grain weight, and grain husk ratio. Genotypes in cluster 4, like Tillenayagam, Chettiverpu, Mattekar, Thuyamalli, Kartu Samba, recorded highest mean value for the trait seed length breadth ratio. Hence, these genotypes can be utilized in breeding programs for integration of desirable traits in the development of high-yielding cultivars. Thank you, ma'am.
Next, Aarti.
Yes, ma'am. Nice, ma'am.
Okay, you can start your presentation.
Okay, ma'am. Good evening to one and all present here. Myself, Aarti. My poster presentation is about study on variability, heritability, and genetic advance in wild rice magic population for yield and yield-related traits under sodic soil condition. Genetic variability in a breeding population is bedrock for any crop improvement program. The study was carried out using 98 wild rice magic lines in randomized block design with re-replication. The study indicated that high PCV and GCV was observed for flag leaf length, number of productive tillers, number of grains per panicle, flag leaf area, and grain yield per plant. High heritability coupled with high genetic advance was recorded for the traits taste 50% flowering, plant height, flag leaf length, and grain yield per plant. The selection method of breeding can be used for improving these characters. Hence, the encountered lines with sufficient variability in magic population can be used to develop desirable lines with high yielding sodicity tolerance. Thank you, ma'am.
Next is Molly. Molly, are you connected?
Yes, ma'am. Can I present now?
Are you connected with us? Can you hear us?
Ma'am, can I present?
Yeah, you are audible and you can start presenting.
Good evening, ma'am. I'm going to present about synthesis and characterization of various iron helates using organic and amino acid as helating agents. Micronutrient deficiency are major concerns in crop production of present-day agriculture program. Helating is nothing but metal ion combined with various helating agents, namely organic acid and amino acid used for helating agents.
By using this yielding agent, we can, when compared to inorganic fertilizer, low quantities are enough. These yielding micronutrient fertilizers are easily absorbed by plant and also easily translocated within the plants. And these yielded micronutrient fertilizers are easily compatible with most pesticide and pesticides also. Here, the yielded micronutrient fertilizers are synthesized based on 2:1 molar ratio, and here majorly 20 number of iron helides are synthesized, and these properties are listed here. By using this iron helides, we are characterized pH, EC, color, hygroscopic, solubility, and iron content. And also we are manipulated the structure by using Schrodinger master software. And among the various iron helides, ferrous and solubility and the lower cost of preparation and the iron content. Thank you, ma'am.
So next is Santosh Kumar.
Yes, ma'am.
Okay, you can start.
So, good evening, one and all present here. Now I'm going to present my topic on termite mound soil as an alternative soil amendment for enhancing food production. For several centuries, termite soil has been used as a amendment in soil to increase the nitrogen and phosphorus sources, which is already present in the soil. This is an ethnobiological fact. To prove this fact, we collected soil samples from 3 different agroclimatic zones. About 10 samples are collected, and the soil physiochemical properties such as pH, EC, organic carbon, as well as soil enzyme activities such as cellulase, dehydrogenase, phosphatase, which are the indicators of microbes in the soil, are calculated. So from the results, it can be implied that compared to the adjacent soil, termite mound soil has enhanced activity of soil enzymes as well as and it also provides a better physiochemical properties such as aggregation of soil, which can be brought about by the foraging activity of the termites, such that the termite soil can be used as a soil amendment, which enhances the food production and by decreasing the chemical fertilizers. Thank you.
So next is Sujita. Sujita. Next is next Harinandan.
Yes, ma'am. Hello.
Is it audible for you?
Good evening to one and all. Yes, ma'am. Good evening to one and all. And my topic is usability of monthly extended range forecast service to predict the maize yield using DSAT crop model over Erode district of Tamil Nadu. Extended range forecast service means the IMD deliver the ERFS in 30 days interval for a state in single values. So this can be used in collaboration with DSAT crop model to predict the crop yields, and this can be done by disaggregation methods. On doing disaggregation, we found that rainfall data of disaggregated weather is correlated very excellent condition with the observed values, and the maize yield simulated by the DSAT model showed 29.1% variation from the actual yield data collected from the district, and it shows the not usable category, but it can be improved by improving the resolution of the. Okay.
Thank you. Rohit, are you online?
Yes, ma'am. Yes, ma'am. Can I present, ma'am? So good evening to all. Today I'm going to present my topic on volatilization, loss of nitrogen and various coated urea in rice and the Tamarabani River Basin. So as we all know, in the event of high input agriculture, more emphasis should be given on nitrogen use efficiency, especially nutrient use efficiency, especially nitrogen use efficiency. That is very low and around 50%. So that is due to several losses, and among them, ammonium volatilization is a major loss. So to study the effect of the coated materials with various levels on the growth and yield of rice crop, field experiment was conducted, and also incubation experiment was carried out to study the volatilization loss. And from this, what the results what we have got is ammonium volatilization was more under uncoated urea compared to the coated urea. So this is because coated nitrogen fertilizers, they will release nitrogen slowly. So because of that, losses are also reduced. And in 100% STCR recommended nitrogen among the different coated materials, fly ash coated urea, in that the volatilization loss was much lesser. And its effect on the yield is that in fly ash coated urea, 100% it gave higher yield compared to the uncoated urea. So from this, I've concluded that 100% STCR recommended N as fly ash coated urea significantly produced more high grain yield. Same way, volatilization loss was also lower in coated urea, especially fly ash coated urea. Thank you.
Dear students, so on behalf of the SPGS, we sincerely
Parthiban is online. Online
every
will come.
student who have presented online, and also those some of the students are not able to present online. So we have evaluated the poster, and some presentations are really nice, and so it shows your sincere effort in making the poster. But overall, we have observed that you have to concentrate on the font size, and you have to present your conclusion and methodology, everything in bullet points, not in paragraph. So you please improve it. With that, we conclude this session. Thank you, one and all. And also, I thank Dr. Sivakumar and Sarah for being the session members. Thank you.
We are also very grateful, ma'am. Thank you so much for helping us in successfully completing this poster presentation. Thank you. So thank you. Sign off. Thank you so much.
Leave the meeting.
நீங்க அதெல்லாம் க்ளோஸ் பண்ணிடுங்க